Welcome to the 12th and final lecture in this class, Marine Ecology, in which we talk about community ecology. So we move from ecosystem to community ecology, from flows of energy and nutrients to species in a local community. The kinds of species, their function, their roles, their abundance, and the number of species at any particular location. So when you're on the beach out snorkeling, you see some fish, you see some invertebrates. What determines which species are there, which species are not there, and how many species are there in total? So this is subject of biodiversity, all the patterns of biodiversity and what causes patterns of biodiversity across the planet. And how is biodiversity related to the fitness of organisms? The emergent properties of the ecosystem that we look at in ecosystem ecology are properties related to the flow of energy and the cycling of elements through the community. And we study that using a system model of the community made up of compartments and quantitative flow rates of energy and elements through those compartments. In community ecology, we look at the emergent properties that are related to the kinds of species that exist in the community and their functional properties and their relative abundance. The kinds of species include taxonomic information, so species, genus, and family. Functional properties include types of behavior, how they use the environment, habitats they occupy. For example, predators may wait in hiding and ambush their prey, or they may actively cruise and search for prey. Predators, when they find prey, may actively chase them, or they may just wait for the prey to get close. So community ecology wants to understand how and why these collections of taxonomic species and their functional properties come to be assembled together at any particular time and place. So, in other words, wants to understand what determines community assembly. So, ecosystem ecology has been called community function, with the idea that communities are analogous to organisms. And a holistic understanding of a community requires us to understand energy flow and nutrient cycling, just like understanding an organism requires us to understand metabolism, meaning all the chemical changes and requirements that keep it alive and functioning. So community function, in other words, ecosystem ecology, is in contrast to community structure, which looks at the kinds of species present, how many species of each kind is present, and the relative abundance of each species. So this is just some common terminology used with common definitions. Where does community ecology fit in Darwin's scheme for the struggle for existence? Well, community ecology fits into the final outcome of the struggle for existence. Species are matched to their environments, and most species are nearly absent from nearly everywhere, but occur only in the locations where they are adapted. So, to the conditions and resources that specifically exist at that location. So, community ecology wants to understand each community as a collection of mutually compatible adaptations. So each species is adapted to the abiotic conditions present at a location and also adapted to the biotic conditions presented by all of the other species that exist at that location and all those interactions that are going on. So we can add to our little table of emergent properties of organisms, populations, and communities. We can add to this species diversity and patterns of species diversity. in communities, an emergent property of biotic communities. And the species diversity, patterns of species diversity, result from all of the outcomes of these population interactions at a particular location in a community, which result from all of the fitness relationships among the species and how they affect each other in terms of survival and reproduction. So ecosystem ecology is usually a sort of more of a holistic field and approach in which you compartmentalize communities and study flow of energy and nutrients through those compartments. Community ecology tends to be more reductionistic, but can be holistic. It ignores energy and nutrients and studies species abundance in space and time, and understands that in terms of a small number of interactions, usually among species in a community. Okay, and this ecosystem ecology is often called the function of communities, studies of the function of communities, whereas this particular approach is described as looking at the structure of communities. Okay, fundamental question in community ecology: what determines the species present at any particular time and place in a community? Talk about the number of species, the types of species, taxonomic and also functional, and the relative abundance of each of those species. The basic model of community assembly. So this is basically the question of what determines community assembly. And the basic model considers 4 conceptual factors that determine what species are present. First, you have the total species pool, and this is the total set of species that can be considered as potential members of any local community. So this really is a theoretical set of species, a hypothetical set based on circumstantial arguments. For example, the species pool of fish that applies to any particular beach location in Zadar might be all species in the Adriatic Sea. That might be the species pool. Or alternatively, it might be all species in the Mediterranean Sea, as each species potentially could migrate to that beach location in Zadar, at least in theory. But not all the fish present in the Med or in the Adriatic are going to be at a particular beach in Zadar at any particular time. The vast majority won't be there at any sampling time, anytime you're snorkeling there. So what is preventing the vast majority of species in the species pool from being present in any local community? What's preventing it? Well, obviously the number 2 factor determining the species present is dispersal constraints. So some species in the total species pool are prevented from dispersing into the local community. Whatever prevents dispersal is called dispersal constraint. So this simply might be just distance. If some species becomes absent from a local community, the closest individuals of that species that could migrate in to replace that absence could be hundreds of kilometers away, and therefore it might take considerable time for that species to reappear, to migrate, to travel into that community. So the reason that species is absent is because there hasn't been enough time for that species to migrate in. Another dispersal constraint besides distance could be a geographic or oceanographic barrier. For example, if dispersal is by larval stage and larvae disperse passively in the ocean currents, the currents might not be in the right direction to allow larvae to disperse in and replace the lost species. For example, if dispersal is by larval stage, and larvae disperse passively in ocean currents. The currents might not be in the right direction to allow larvae to disperse in and replace the lost species. And the third factor is environmental constraints. So this is the idea that species are adapted to a particular habitat type, and this habitat type allows refuge from predation and has a steady supply of limiting resources for the species and allows any social interaction necessary for mating and rearing young. The reason some species is not present at a particular location may simply be that the habitat that the species is adapted to is not there. Remember, the result of the struggle for existence, species are absent from where they are not adapted. So habitats that require different adaptations, for example, are benthic habitats such as seagrass, macroalgae, bare sand, bare rock. A fish species might not be present in a bare sand community because it's not able to easily escape predators there, as for example by burying itself in the sand or making use of cryptic body coloration that resembles sand, or an invertebrate such as a mussel has no hard substrate to attach to. And the subset of the total species pool that is capable of living in the local habitat is called the habitat species pool. The subset of the total species pool that's capable of living in the local habitat and capable of dispersing to that habitat is called the ecological species pool. And by habitat, this is probably not a good term to use here. Probably a better term to use, as we've seen, is the niche. So the niche species pool is probably a better term to use. So species are restricted to their ecological niche. And that would be the fundamental niche here. And the fourth factor is internal dynamics. Even if a species is in the ecological species pool, namely is not prevented from being in the community by any of geographic constraints or environmental or fundamental niche constraints, because it's adapted to that niche or habitat and is capable of migrating there, nevertheless, it might not be present in that community because of internal dynamics. In other words, this might not be the realized niche. For example, it may be an inferior competitor, the species already present, and so is competitively excluded from the community. Or it might be a prey species excluded by a predator already in the community. Or there may be diseases in the community for which it has no immunity. So community ecology is to a large extent a study of each of these constraints: total species pool, dispersal constraints, environmental constraints, and internal dynamics that prevent a species from being present within a particular community at a particular location at a particular time, and the net effect of all these constraints on the biodiversity of the community, and on how that biodiversity changes at different locations at different times because of various factors related to environmental constraints, dispersal constraints, and internal dynamics. And all of these factors are important for basic reasons, just understanding, and all these questions are important from a basic point of view, just a basic knowledge, basic understanding of why communities and populations and organisms are the way they are. And they're also important, of course, for practical reasons, for economic reasons, resource conservation, and for sustainable use of those biotic resources, and also for protection, environmental protection, for managing and preserving biodiversity at any location, for all sorts of personal, human, economic, and social reasons. Let's look first at dispersal constraints. Dispersal constraints were first quantified within a mathematical theory in a theory of island biogeography. Insular zoogeography is what it was first called, but we now call it just island biogeography, which was developed originally by Robert MacArthur and E.O. Wilson, two famous ecologists, in a paper published in the journal Evolution. So this theory gave several testable predictions relating to the number of species in a community, to the area occupied by the community, to the distance of the community from the total species pool, and the size of the island the community is located on. Island biogeography is an equilibrium theory. In other words, it predicts certain patterns at equilibrium, namely when counteracting processes reach some balance that is relatively stable. So first we imagine an island, and the island could be a real literal island in the ocean, or it could be a figurative, sort of metaphorical island, as for an example, an island of habitat. And this is not really metaphorical, it is still a real island. It's a spatially restricted area where the realized niche occurs for a particular species. And second, we ask the question, what determines the number of species present on that island at any time? And to answer this question, we imagine that during any time interval, day, month, year, some number of species will colonize the island and some number of species will go extinct. Colonization simply means that at least one individual arrives on the island belonging to a species that is not yet on the island. And extinction simply means that the last remaining individual of a species dies, and therefore the species is no longer on the island. So this is similar to the metapopulation model that we talked about in the population part of the course, but we're moving up from the population level of organization to the community level of organization. And instead of looking at the presence and absence of one particular population, we're looking at all the populations together of all of the species on this particular habitat or niche island. Okay, so then we imagine colonization and extinction rates, in other words, the number of colonizations and extinctions per unit time, and how these two rates depend on the number of species on the island. So first, how does the colonization rate depend on the number of species on the island? Well, if there are no species on the island, then the colonization rate will be high. Because every or nearly every individual that arrives on the island will be a new species and therefore a colonization. Conversely, if there are many species all on the island, so this x-axis is the number of species on an island, so as the number of species goes up, if there are many species already on there, then an individual arriving on the island from outside is likely to belong to a species already on the island, so is not a colonization. So the colonization rate, which is the y-axis here, of an island depends on the number of species that are on the island already. In terms of probability, we could say we have two urns, or two bowls, two jars, one containing the total species pool, and the other containing some subset of the total species pools, representing the island. If we draw a species at random from a potential colonist from the species pool, what is the probability that it is of a species not present on the island? That probability, of course, will be smaller and smaller as the number of species on the island increases. Second, how does the extinction rate Now we're considering the y-axis, the extinction rate, the same units. How does this rate depend on the number of species on the island? Well, if there are no species on the island, obviously the extinction rate is 0. Let's say each species on the island has a small probability of going extinct during any time period. So if there are a large number of species on the island, then the extinction rate will be higher, since each of a large number of species could go extinct during that time period. So there are two curves we can draw in a plot of colonization and extinction rates as a function of number of species on an island. The colonization curve has a negative slope, might be curved in some way. The extinction curve has a positive slope. The extinction curve starts at 0, and the colonization curve starts with some positive number and ends at 0. So let's imagine an island with no species. What happens? Well, colonization rate will be higher than extinction rate. So the number of species on the island increases over time. Now let's imagine an island with a large number of species. Extinction rate will be higher than colonization rate. So the number of species on an island, on a large island, will decrease over time. What's the equilibrium number of species on the island? It's the number of species where the two curves cross. In other words, it's the number of species for which the extinction rate equals the colonization rate. So if the extinction rate equals the colonization rate, then there's no change in the number of species present on the island. The actual species might change randomly, although there is continual change, so in the kinds of species present. Now imagine how extinction and colonization play out against the size of the island. So we've looked in terms of the number of species on the island. We've got this nice equilibrium here, makes sense intuitively. What if we plot instead the size of the island? And remember, we're talking about a real island or a habitat island, a geographic island, spatially restricted geographic island of habitat. All right, here we have the same plot, and we're just drawing two different curves of the extinction rate for two islands of different size. So a large island may contain more species for a variety of reasons. One reason might simply be that the extinction rate is higher for smaller islands. Why should the extinction rate be higher for smaller islands? Well, because maybe population densities are smaller on smaller islands, and therefore populations are more vulnerable to demographic stochasticity. If the extinction rate per species is large on smaller islands, then the extinction curve will be higher on smaller islands. So as the number of species goes up, the rate of extinction is higher in both islands, but the rate goes up faster on smaller islands because each species has a higher probability of going extinct on a smaller island. So if the extinction curve is higher, then that means where it crosses the colonization curve is farther to the left, which means that fewer species are expected at equilibrium in smaller islands. So this theory gives us a testable prediction. Testable prediction from the theory of island biogeography. Smaller islands should have a smaller number of species at equilibrium. Well, do they? Well, that was one of the purposes of this paper that MacArthur and Wilson wrote. They wanted to test this hypothesis by counting the number of land and freshwater bird species on various islands in the Malay archipelago in Malaysia. They found a huge difference in the number of bird species dependent on island size. The largest islands had 100 times the number of species that small islands had. So it was a nice confirmation of the theory, but Does this mean that island biogeography is correct? No, there are other reasons for more bird species on larger islands, just a lower extinction rate curve. It could simply be that there are more habitats on larger islands. But the first test of a theory is to see if its predictions hold true, and they do. If the predictions are not met, then we can discard the theory. But if they are met, all that means is that we can't discard the theory yet. It could be true. But there are other theories that can explain the prediction. They haven't yet been ruled out. So we can continue testing. If it is really colonization and extinction dynamics that are responsible for the number of species on an island, then we would expect that islands near the mainland have more species than those farther away from the mainland. And this is because of differences in the height of the colonization curves. So if you're on an island closer to the mainland, then most likely colonization rates are going to be higher because migration of individuals will be more frequent over shorter distances. So naturally, the rate will be higher. So here we have the same extinction curve, but two different colonization curves. One curve, which is higher for near islands, islands that are near the mainland, near the species pool, and far islands that are farther away from the mainland, the source of the colonists. And MacArthur and Wilson tested this hypothesis and confirmed it for the Moluccas, Melanesia, and Oceania islands. And MacArthur and Wilson tested this hypothesis and confirmed it for the Moluccas, Melanesia, and Oceania islands, showing that the greater the distance of an island from New Guinea, the lower the percent saturation of the island in its total number of bird species. This seems to be a strong evidence for the effect of colonization on total species on an island, since distance from the mainland seems unlikely to be confounded with any other possible explanation, such as the number of different habitats on the island. And this hypothesis was also tested and confirmed in a manipulative field experiment by Simberloff and Wilson, in which they counted the number of arthropod species, so insects, spiders, and crustaceans, on small mangrove islands in the Florida Keys of the U.S. Mangrove trees grow in seawater, and their roots trap sediments. And over time, a mangrove tree will become a small island. There are millions of mangrove islands in South Florida, a perfect situation for testing predictions of island biogeography. So the researchers looked for small islands that varied in their distance from a larger source island from which the small islands received colonists, insects, spiders, crustaceans. So they used methyl bromide, a chemical used to kill insects, to fumigate the islands, killing all the arthropods on the islands. So you had a completely clean island, clean of all arthropods. And then they monitored the islands over time afterwards, after fumigating them, to count the number of species that colonized the islands over the next year. They put up a bunch of different types of traps. And they found that islands closer to the source of colonists had a higher colonization rate, and eventually a higher equilibrium number of species than islands farther from the source island. So this is a plot of their results. Before fumigation, this was the number of species present on the nearest island and then progressively farther away. So they found that as you got farther away from the source island, the number of species was smaller. And then at time zero, they fumigated all these islands, and then they watched how long it took over several months for colonization to occur and create a new community of arthropods. And they found that the nearest island eventually ended up with the largest number of species, followed by the next, and the next um, nearest island, farthest away island having the fewest species after about 12 months. So if they continued monitoring past the 12 months, then perhaps all of the islands would eventually reach their original number of species. All right, many such studies of island biogeography have shown, as general result, that there is a fundamental mathematical relationship between the total area of a habitat island and the total number of species found in that island. This is called the species area relationship, and this relationship is an exponential relationship similar to the allometric relationships between the sizes of different parts of an organism. So this relationship is just S, the number of species, also called the species richness on the habitat island, is equal to A, which is the area of the island in hectares or square meters or whatever, and C and Z are two constants that vary depending on the taxonomic group of species and the type of island. This is called the species area curve or relationship, and it has been considered one of the most important relationships in community ecology that can be universally applied. So Durrett 96 said, "This is the most profound empirical observation in ecology. No relationship is more informative for understanding how communities are organized." And here's an example of a beach habitat. So beaches are, a nice example of an island habitat, geographically restricted habitat. And McLaughlin and Dorvlow studied the macrofauna in around 100 sandy beaches all over the world and counted the species richness of marine species and related this to the width of the beach. Width is sort of a proxy for size of the habitat island. Beaches vary in their width because the tides in different places vary in their amplitude. The width of the beach is just the distance from the ocean to the land, perpendicular to the shoreline. So the tides are different, so the width of the beach is different. It's an intertidal area. Beaches vary in their distance from the amphidromic points of the different tidal components. So they looked at species, clams, mole crabs, various types of beach hoppers, worms, shrimp, and so on, living on these beaches all over the world. And they plotted the relationship between marine species richness found on a particular beach and the width of beaches all over the world. And convert to the log scale, we have something that looks close to a linear relationship. Could be, although there's some indications of these 0 points, which are a little bit concerning, maybe outliers. So if you fit the straight line on the log relationship, you get this right here. And so we've got, this is our species area relationship for sandy beaches worldwide. S is equal to 1.22 times A to the 0.5 power. In this case, A, the units of A is width of the beach in meters. Okay, another type of island habitat is the coral reef. So coral reefs are geographically restricted habitat islands. And you can do the same sort of study with coral reefs. And this is what Chitaro did. Calculate the species area relationships for coral reef fish on one of the U.S. Virgin Islands. And here's the curve that he plotted between the log of species richness, the number of reef fish species present at a particular reef, and the log of the area in meters squared of the particular coral reef. And he's got two curves for different types of reefs that are just isolated patches and reefs that are part of a larger continuous reef. In any case, the point is that we've got this nice species area relationship as predicted by island biogeography theory. The causes of the differences between the two types of reefs still unclear, but apparently is related to the number of distinct microhabitats available on the two reef types and how those spatially vary within the two reef types. So obviously other factors than just size influence the number of species present, but we still have this nice size richness relationship predicted by the theory. Okay, so we've got this nice species area relationship. What are the possible causes of it? Well, there are causes other than those causes hypothesized by MacArthur and Wilson in the original island biogeography theory. So, first possible reason, this could just be a statistical artifact. If there are more individuals per species on larger islands, more abundant species on larger islands, then you're more likely to sample each species and observe it on a larger island than on a smaller island. So this phenomenon is just called rarefaction. So it's just a statistical artifact. The second explanation, as area increases, the number of different habitats also might increase. In other words, habitat diversity. And if different habitats tend to have different species because of their different adaptations, then a higher area will necessarily contain a higher species richness, independent of anything else, colonization or anything else. Three, if a higher area generally contains more individuals per species, then populations in larger habitat islands will experience less demographic stochasticity and therefore have a lower risk of extinction. So this was hypothesized by the original model, the original MacArthur and Wilson island biogeography model was based on this prediction right here. And fourth, if species occur generally as metapopulations with many subpopulations experiencing occasional migration and colonization and extinction, remember our blinking Christmas tree light model, then larger habitat islands might have more subpopulations per metapopulation and therefore higher colonization rates and lower rates of subpopulation extinction. So another reason for the species area relationship is metapopulation dynamics. And so this was also considered in the original model. Okay? So these are all the possible explanations for the species area relationship. All right, how do we conceptualize this in terms of our pools? Well, we've been looking at the question as to why we see a particular assemblage of species at a given location, at a given time, in other words, a particular community, and considering the four species pools, a total, geographic, habitat, and ecological species pool. Island biogeography provides us with a mathematical treatment of dispersal constraints and the geographic species pool right here. As, for example, the relationship between the number of species on an island and its distance from the mainland, which can be considered the total species pool. However, some of the relationships seen in island biogeography, for example, the species area relationship, might not be due to dispersal constraints, but to habitat or niche constraints, environmental constraints. In other words, the number and type of species in a given community certainly also reflects the number and type of habitats or niches at that location. And there's a huge number of studies published showing the relationship between habitat diversity and species diversity in different types of communities. Just for a couple examples, so Chitaro again looked at the total microhabitat richness of coral reefs, so patch reefs, isolated patches, and reefs that are part of a continuous larger reef, and found that for both reef types, both habitat richness and the richness in algae and seagrass species increased with the area of the reef, and that 50 to 80% of the increase in species richness of both types of with area was caused by an increase in microhabitat richness. And Darnell, in this classic study in 1970, found that the number of fish species increased along a river drainage from headwater springs to the river mouth. And that increase was associated with increased diversity in food sources of those fish. At Headwater Springs, the only food source was detritus, this brown color here. And as the stream became larger and larger downstream, there were more food sources and more fish species. Larger streams contained arthropods, algae, and vascular plants, and other fish. So the most diverse food sources were at the river mouths, which is also where the largest number of fish species were found. Okay, so habitat diversity is an important factor that may be associated with size. And it's another factor that we can look at separately from size. So let's take a look at habitat variation and how to quantify habitat diversity in communities. So habitat constraints are analyzed and studied in the process of gradient analysis. Gradient analysis looks at gradients, continuous changes in communities across a spatial dimension or in time, and gradients in space are then related to gradients in habitats within those communities. So gradients in time have a special name. Those are called ecological succession. And these temporal gradients are related both to colonization constraints and to internal constraints. So for example, competition and predation, which we'll also consider. But for now, let's consider gradients in space and consider how communities change along a spatial dimension. And we want to eventually relate that to habitat. So that spatial dimension, for example, can be depth of the ocean. We can consider invertebrate communities from shallow to deeper locations. Okay, so one possibility. So we're doing a gradient analysis of the invertebrate community along the depth variable. The gradient could also be by wave exposure, for example, how the species in an algal community might vary along transects from high wave exposure to lower wave exposure. So there are many possible gradients where we could analyze communities. So for example, by depth, wave exposure, light, salinity, current speed and direction, temperature, anthropogenic disturbance, from low disturbance to high disturbance, size of sediment particles, aspect of exposure of the shoreline along compass points. So how the community changes, whether the shoreline is facing east, west, north, or south, and so on. And if we do this, we can make a plot like this of abundance of different species, and these are the different colors of the bands here, against whatever environmental gradient we can be looking at. And if you look at these two plots, you can see that you can conceptualize two extremes of community variation along a gradient. We can see sharp boundaries in many species occurring at the same time as we move along the gradient. That's this top plot here. Or we could see no sharp boundaries in many species occurrence. And so in this former case, where you see sharp boundaries in many species occurring coincidentally at the same point in the gradient, this is called closed communities. There are sharp boundaries separating different whole communities containing different groups of species, and such sharp boundaries are called ecotones. In other words, on one side of the ecotone, you have one collection of species in the community, and on the other side of the ecotone, you have a different collection of species in the community with little overlap in species. So the communities are called closed because it appears that they're closed to migration across the ecotone. If you're in this part of the gradient, then you cannot move to this part of the gradient. The community is closed to you for some reason. So in open communities, in contrast, there is no ecotone. Species are at different locations along the gradient, but different species appear and disappear at different points along the gradient rather than all at once at an ecotone. So, I mean, this community is different from this community, but there's no sharp distinction between the two communities. Right, so how do we quantify this? This is done in the process of community ordination, in which in R you can do it with the package vegan very easily. And in community ordination, the data is the community or species matrix right here. And we want to measure the overall similarity in species occurrence or abundance at each of the several sampling locations. So the columns here are different species. So species 1, species 2, 3, 4, and 5 in the column. And the sites here are locations where we do the sampling. So each location we can think of as a different community. So sites could also be communities. Or they could be, sub-locations within a same community that varies. It's just a semantic difference. So in the entries, each of these entries in each cell here is a relative abundance of each species at that site, sampling location. And so these are relative abundances. So they all sum to 1 or 100% at each location. So the question then is, how similar are the two communities found at two sites, any of these two sites compared? Well, they are similar or different in the identity of species at those sites, and they are also similar or different in the relative abundance of those species. How do we take those similarities and differences in each species and calculate some overall level of similarity between the two sites? There are many ways of measuring overall similarity or difference, and Vegan has about a dozen different measures that you can use. They all give slightly different results. And we'll illustrate one commonly used method for measuring the similarity between two communities, which is the Bray-Curtis similarity between two communities. To calculate the Bray-Curtis similarity between two communities, you consider each species present in one or the other community in turn, and for each species you ask what is the relative abundance in each of the two communities. And the relative abundance of each species in a community will add up to 1. And you find the lowest of the two abundances, and you write down that number for each species. For a species that is present in only one of the two communities, its lowest abundance is 0, and therefore does not contribute to the similarity. Therefore, the Bray-Curtis similarity is just the sum of the lowest relative abundances for each species that occurs in both communities. And this number will always be between 0 and 1. If the two communities have no species in common, then the Bray-Curtis similarity will be 0. If if two communities have all species in common, and they all have the same relative abundance, then the Bray-Curtis similarity will be 1. So the Bray-Curtis similarity is very simple to calculate and understand, and can be inverted to measure the difference between two communities. So the BC Bray-Curtis difference between two communities is just 1 minus the BC similarity. So the difference is also called the distance or dissimilarity between two communities. So you can calculate the similarity or dissimilarity between all possible pairs of your study sites. And once you have the BC measure for all possible pairs, you can put that in a matrix that is called the dissimilarity or distance matrix. And that distance matrix is then used to plot each community on a map called the ordination map. And the map obviously is not a real map of the geographic location. It's a map in species space. But this map tells us how different each community is overall in the identity and abundance of species in the community. And the physical distance between two communities in this ordination map is roughly equal to the overall distance in Bray-Curtis units between the two communities. Okay, so let's do a comparison for all possible pairs of sites in their Bray-Curtis similarity and dissimilarity. So how similar are communities A and B? Well, we go through the list and find the lowest relative abundance of each species in these two communities. So for the first species is 0.2, and we have 0.3, 0.2, 0, and 0. So 0.2 plus 0.3 is 0.5, plus 2 is 0.7. So the similarity between A and B is 0.7, and the dissimilarity is 1 minus 0.7, or 0.3. That's all it is. For B and D, okay, B and D, we've got 0, 0.1, plus 0.2 is 0.3. There we have it. 0.3 is similarity, and 1 minus 0.3 is 0.7, and that's dissimilarity. So these are Bray-Curtis similarity and dissimilarity. And dissimilarity is also called distance. So these would be the Bray-Curtis distances. All right, now that we have these dissimilarities, we want to plot each of these communities, A, B, C, D, on a map, on a two-dimensional map. And that's called an ordination, a community ordination. And to understand this idea better of an ordination, think of a real geographic map, say of Europe. And let's say we have no idea what this map looks like, but we do know the rough distance, dissimilarity, between every pair of cities. So we've got cities A, B, C, D, and we know, and these are the distances, the geographic distances as the crow flies between each pair of cities. If we know all these distances, we can create a distance matrix, which with each city as a row, and each city again as a column. And the entries of this matrix are the distances, the pairwise distances between each pair of cities. With this distance matrix, we can create a real geographic map showing each of those cities and their positions relative to each other. We could ask R, using the Vegan package, to create that city map of Europe, knowing only the pairwise distances. And this would be simple to do. And that's exactly the same thing we're doing in creating an ordination map of communities, only we're not looking at cities and real distances in kilometers. We're looking at communities or sampling sites and these abstract distances in species abundance between the communities in Bray-Curtis units or some other kind of distance units. So the ordination map we're creating has communities plotted as points, A, B, C, and D plotted as points as if there were cities in a map. And a community's location indicates its Bray-Curtis distance or difference in species composition from each of the other communities. Okay, so let's do the ordination. Let's plot each of these communities on a two-dimensional map as if they're cities on a map. And their distances between them are exactly each of these. So that's the challenge, to plot a map, plot A, B, and C, D on a two-dimensional map, whereas if you measured the distance between each possible pair, you'll get exactly these numbers on this two-dimensional map. All right, let's start by doing this on a one-dimensional map. Okay, so I've got just this one-dimensional, this line, and I'll just put A here, and we know that B is 0.3 units from A, so I'll put B right here. And we know C is 0.4 units, C is 0.5 units from B, so I'll put C right here at 0.8. And we know that D is 0.4 units from C, so I'll just put D right here at 1.2. All right, there's my ordination. I'm done. Or am I? Does this make sense? Okay, well, let's see. What is the distance between A and C supposed to be? Well, that's supposed to be 0.6. Is this 0.6? Nope, it's 0.8. Oops, it's not a good ordination, is it? And B and D distance is supposed to be 0.7. Is this 0.7? No, it is 0.9. Not good. And then between A and D, we have, should be 0.7. Is A and D, the distance here, 0.7? Nope. So this is a really, really bad ordination. It doesn't really represent the true relationship, distance relationships among these four sites. So if these were four cities, we would have to say, no, these cities are not arranged along a straight line, obviously. So we need to arrange them correctly in two-dimensional space. So how would we move each of these points along another dimension so that their distances, all possible pairwise distances, are exactly as we have written them here? How would we do that? Well, it's not an easy problem to solve. That's why we let a computer do it. So R does this in a package called MetaMDS. It maps communities based on a dissimilarity matrix. So the way R does this is that we adjust the position of each community so that, and it just changes the position by trial and error until it gets a closest match as possible to each of these pairwise dissimilarities in the distance matrix. Okay, so I put these numbers through the MetaMDS package in R and got this nice map or ordination of these four communities. So do the dissimilarities hold here? Well, here are the units right here on the axes. So A and B looks like it's about 0.3 units. They look like they're 0.3 units apart from each other. B and C looks like it's about 0.5. And C and D looks like it's about 0.4. A and D, it's exactly 0.7. A and C is exactly 0.6 as it should be. And B and D is exactly 0.7 as it should be. So R has found a nice solution to this problem of mapping the four communities in two dimensions in a way that the distance, all the pairwise distances on this map, are equal to the dissimilarities that we found for all the pairwise comparisons of the communities. Dissimilarities in species abundance, relative abundance. Okay, so this is how we picture communities with an ordination plot like this, which tells us how different and how similar different communities are, or different sampling sites are. All right, so how does this tell us what the habitat or niche constraints are and what the role of habitat or niche constraints is in determining how similar or how different two communities are? How can we use this to test hypotheses about habitat or niche constraints in causing the assembly of species in a community? Well, right now we have no idea what those habitat constraints are. All we know is that the communities are different from each other or similar to each other according to this pattern. But we have two measures, these two axes of difference. And the existence of these two axes allows us to test hypotheses about habitat constraints. So for example, this x-axis that's separating the species along the horizontal distance might be correlated with some important environmental gradient like temperature or depth or salinity. And this axis might be correlated with some other important environmental gradient like wave exposure or aspect or sediment or currents. And so if we can find some habitat or niche dimension that is correlated with these two axes, then we have potentially explained the major variation among these four communities and explained why they're similar or different from each other. So this x-axis might turn out to be a temperature gradient, so that A and B tend to occur at lower temperatures, and C and D tend to occur at higher temperatures. And this y-axis might be, for example, a salinity gradient, so that A and D tend to occur at higher salinities and B and C at lower salinities. So sometimes just by creating this ordination, we can immediately see patterns relating to gradients in the environment that we already knew about. And then we can use those patterns as hypotheses for further testing in an experiment or some new study. So this is why this is called gradient analysis, habitat or niche gradient. And the idea is that we want to find dimensions of the niche along which communities are organized, communities are assembled, or species are assembled along dimensions of the niches that they occupy. We might not have any idea what those important gradients are when we start this analysis, but once we see the ordination, all of a sudden the patterns start to emerge. And in this case, when we just looked at the differences and similarities, we didn't have this picture in mind. But now that we have this picture in mind, we can see that there appear to be two completely different gradients here. So the first gradient separates A and B from D and C, but it does not separate A from B. It doesn't really separate D from C very much either. But the second gradient does a nice separation of A from B and from D and D from C. So we found two gradients here that are quite different from each other that seem to be important in determining the similarities and differences among these communities in the species that occupy them. And important thing to keep in mind here that we're only looking at two dimensions. We can look at any number of additional dimensions as well, 3, 4, 5, 6 dimensions. Remember, niche is a multidimensional hypervolume. And you can look at as many dimensions as are important. So you can keep adding dimensions. But usually people start out with just two as a first approximation. And if two don't separate the communities very well, you can add a third and a fourth, and maybe you can get better separation. And it's easy to see why this is true, why there can be more than two dimensions that are important to look at. So imagine instead of cities on the surface of the Earth that we're plotting, we want a map of the stars in the sky, and we have a distance matrix between all pairs of stars that we're interested in some region of space. And now we do the same thing. We create a map of those stars with two axes and two dimensions. Obviously, this map will not show the true distances between each pair of stars because two stars might be close together on our map, two-dimensional map, but they may be very far apart in space. So this is because the map is in two dimensions here, whereas the stars are separated into three dimensions. So to make an accurate star map, we need to have three dimensions, not two. The same problem might occur in our community ordination plot. So communities might exist in three dimensions, three important habitat gradients, or three important niche dimensions, and not two, and therefore showing an ordination map in two dimensions might not preserve the Bray-Curtis distances accurately. And you can see that whenever you do this analysis, it gives you an estimate of how good the fit is. That's called the stress. So if the stress is too high, you need to add a new dimension. And that might be because there are three environmental factors that are determining species abundances and not two. If there are more than two environmental factors, then the 2D ordination map will, it's not going to be a good display of the community differences. Two communities or sampling sites might be close together in 2D map, but actually the species compositions might be quite different, because there's a third environmental gradient that separates those communities. It's not portrayed on the map. So whenever we do an ordination map, there needs to be some statistic that shows overall how close the ordination distances on the map match the true distances that were used to generate the map. And different ordination methods have different measures of the fit. So like I said, for NMD, and R, stress is the measure, and others have different ones. And usually it relates to the amount of variation they're able to explain. So this is one of many methods of doing a gradient analysis. We can plot communities on an ordination map to see how different they are from each other, then ask how those differences are correlated with environmental factors or gradients. And so let's look at a few examples of going from a community matrix to an ordination plot. So fish communities in the Novigrad Sea in Croatia. So this is a paper out of our lab done at the University of Zadar, looking at predator-prey segregation, habitat segregation in an Adriatic lagoon, basically the Novigrad Sea. And if we plot the ordination diagram based on fish abundances, we can see that the two axes are correlated. And this is a different type of analysis. This is called detrended correspondence analysis, which, is not a strong consensus among scientists about which method is the best for doing an ordination. Anyway, we can see that the two axes are correlated with the benthic habitat. One axis has unconsolidated sand or sediment habitat to the left, which is U, and vegetation to the right, which is algae, here A, and dense seagrass, macroalgae and dense seagrass. And the other axis is correlated with type of habitat structure. We've got rock and algae at the bottom and seagrass, dense and sparse, at the top. So this particular diagram doesn't plot the actual communities or sampling sites themselves, but rather plots the species in the communities and the habitat types from their correlations with the two axes. So showing the sampling sites themselves doesn't necessarily provide the most interesting information on the plot, which is why we don't show them here. Rather, the species and environmental factors are usually what the reader wants to see. So in the seagrass habitat, dense seagrass habitat, we have these species. Sparse seagrass habitat, we have these species. In the U habitat, we have these species right here tend to occur. In the R, we have these species. In the A, we have these three species, are the most characteristic. And then the species that are closer to the origin here, they basically don't seem to have much correlation with habitat, just the ones out here on the ends of these arrows. So the ordination map is a very convenient and useful way to visualize communities and why they are the way they are. So we can see where each fish species sits in the ordination space and where each habitat sits in the ordination space. You can see visually how each species abundance is correlated with the habitat and vice versa. So it gives an overall picture of how the fish community is organized relative to the bottom habitat, or whatever the niche dimension is that you're interested in, or whatever it is that is organizing these communities. If the communities were also plotted in this graph, we would see that some communities are over in the seagrass region of the plot, and others are over in the algae or rock or unconsolidated sediment region. And these are different communities at different benthic habitats. The ordination map shows how environmental constraints, habitat or niche constraints, might create the habitat pool for the species, might explain why some species from the geographic pool are not at that particular location, because the benthic habitat that they require does not exist at that location. And this is a community level analysis. Of course, we could go down to the organism level as well and ask what these species are doing that adapt them to this algal habitat. Why are they adapted to the algal habitat? What these species do that adapt them to the R habitat, and what these species do to adapt them to the unconsolidated habitat. So we'll find, for example, that they are adapted. So these species, for example, tend to hide cryptically in the sand, and some of them might bury themselves in the sand a little bit to avoid predators. And these species here in the seagrass might use seagrass as cover where they hide in order to ambush their prey. So the predators that are ambushing their prey within the cover of the seagrass. Okay, so you can start to see how a community level analysis can give you insights into organisms as well, which of course Darwin predicted in his struggle for existence. Okay. So the idea of ordination is that purely by measuring relative abundance of species in communities and seeing how those relative abundances are vary at different locations, the pattern that you see as a result can provide evidence for the environmental or habitat constraints that are determining the presence or absence of species at any particular location and how abundant they are. All right, so ordination is not the only way to visualize relationships among communities. There's another common method for visually portraying differences among communities, and this is called hierarchical clustering. So creating basically a tree instead of a map. And the tree is called a dendrogram. So that's just a fancy word for a tree with branches. So a tree diagram where each community is placed on the tip of a tree branch. And so in this diagram, the distance along the branches between any two communities is the Bray-Curtis difference between the sampling sites. The advantage of hierarchical clustering is that it preserves all distances exactly. There's no information lost, as there might be within an ordination plot in only 2 dimensions. So if you don't plot the third or fourth dimension, there's going to be some mismatch between the ordination distances and the actual distances. And the disadvantage of hierarchical clustering is that it's not a map, so it's difficult to see how the differences are sorted along any gradient, any environmental gradient. So it's difficult to see the environmental gradients in this dendrogram in, say, 2 dimensions. And a hierarchical clustering plot can be based on sampling sites, not quite as interesting, or it can be based on the species, which is a lot more interesting. The species are considered again, like European cities, and there are differences between each pair of species based on their relative abundances in each of the sampling sites. So how those species then sort out in clusters can then define different community types or different species clusters, mainly species occurring together in sampling sites. So here we have a cluster of species right here on this branch, and these turn out to be species that tend to occur in dense seagrass. So this is our dense seagrass community right here. And they're hierarchical clusters. And if we look at other clusters, we can see rock-associated species, and we can see sand-associated species. And we can see the difference between dense seagrass and sparse seagrass communities. So this species-based hierarchical clustering analysis can give you an idea of, what species identities are in different communities. So since we're discussing clustering based on multivariate distances, it's worth pointing out that this method has many other uses in ecology. One example is is analyzing the community of species in the gut contents of fishes. So each species of fish has a different community of prey species in its gut, and you can map each fish species in an ordination map or tree diagram showing the overall difference in these gut communities in different fish species. So again, we're looking at Bray-Curtis similarity, percent similarity, and we're looking at each species, and these species are compared based on how similar their gut contents are to each other's. So this is a Pinagar and Palunin study in 2000. So this diagram can be used for a variety of purposes in conservation management or generating testing hypotheses about competition and resource partitioning. Species more similar in feeding requirements are expected to be more in competition with each other if occurring at the same location. So we might expect the community occurrence dendrogram to be negatively correlated with the feeding dendrogram if species tend to organize in communities to avoid competition, or if competition tends to destabilize certain combinations of species. So, for example, you might find that Scorpaena porcus and Sirena scriba tend not to occur together in communities because, look, they basically have a very similar gut content. So they might be in intense competition for food if they occur together. Similar for Chromis chromis and Symphytis oscillatus. So far, we've outlined some work on geographic constraints, dispersal constraints in island biogeography theory, and habitat constraints using community ordination in driving community assembly and controlling what species are present or absent at any particular location. So now what we're going to move to are internal dynamics, which are the final control over the species that are able to persist together in a stable coexistence in a local community. So the species that are capable of persisting together in a community are those able to survive or avoid loss of fitness due to interactions with other species. And those, remember, are plus or minus interactions such as competition, predation, parasitism, disease. And there are many examples of studies of internal dynamics, and many classic examples are marine examples because of the abundance and diversity of species in the rocky intertidal habitat, which is a very easy habitat in which to do manipulative experiments. And it's also very scenic and beautiful if the weather's nice. So you can manipulate one population on the rocks, you can remove it or enhance it, and then you can monitor what effect that removal or enhancement has on other species. So these are exclusion or transplant experiments. So you spend a nice day at the seashore and accomplish some science too. For example, these types of experiments were reported on the rocky coast of Patagonia by Burtness and colleagues, 2006. Found that a kind of mussel was the dominant competitor for space and excluded almost all other species in wave-exposed areas because it was more adapted to wave energy than other species, so internal dynamics prevented many species from persisting in the presence of mussels at all levels of the intertidal, high, middle, and low, in wave-exposed areas. However, in wave-protected areas, the mussels could not survive the upper intertidal due to heat and desiccation. Wave spray no longer kept them cool and wet, and they dried out and overheated. They also couldn't compete in the lower levels because coralline algae were more superior competitors for space, where wave energy was low, and coralline algae could only survive in the lower levels. In the middle levels, the mussels still dominated in wave-protected areas. So this is an example where competition was shown using removal experiments, just like what Connell did, to prevent species from maintaining populations in a community. And competitive ability was related to the conditions the local community site. In this case, the main condition was wave energy. So it's a physical factor that was primarily responsible for these differences and these different interactions and interaction constraints, preventing the mussel from making it in some areas. Another example in the Novigrad Sea of Croatia, the same paper as before from our lab, we found that fish predators and prey were segregated into different habitats on average, although there was some overlap, obviously, or nobody would be eating each other. Ambush predators tended to occur in seagrass habitat, where they could hide under cover and wait until prey fish came nearby before striking, while prey fish species tended to occur in unconsolidated sediment habitats or at the edges between sediment and rock or algae. So this is a plot of predator density against prey density, and these are the habitats. So we have high predator density in seagrass, dense and sparse seagrass habitats, and low predator densities down here in the algae and rock habitats. And you can see the highest prey density is up here in the unconsolidated sediment habitat for the most part, in some cases some rock and algae. So you see a very nice and very clear and distinct separation between predator species and prey species. So this is an example where predation seems to be excluding prey species from some types of communities, in the seagrass, for instance. And the effect of predation might be a learned response from prey experiencing aggression or injury, or might be an innate fear of smells from certain predatory fish, certain types of habitats. So many studies have uncovered the importance of predation in structuring ecological communities, not only in determining the abundance or presence absence of individual species in the community, but also in controlling emergent properties of communities. And one important emergent property of communities impacted by predation is the species diversity or species richness. So we talked about species richness already in conjunction with island biogeography theory. Now we discuss richness and diversity and their relationship in some more detail. Species richness and diversity, or more generally biodiversity, is one of the most important concepts in community ecology and the study of community structure, as it's basically the center to theories of community stability and is central to conservation and management, basically. So let's consider the difference between species richness and species diversity. So there are two ways to measure the overall biodiversity of a location, or the idea of total number of different types of species. And these two ways are richness and diversity. So in both cases, keep in mind that the kind of species can be two different ways to assess difference. The first way is just a taxonomic designation, the genus of species. But this can be any taxonomic level. You can also look at the richness of or diversity of phylum, class, order, family, genus, species, or subspecies. Because that's the taxonomic definition. The other way is in terms of function. Functional type is usually the type of predator, but it can be anything really. So in other words, how it searches for prey, how it captures prey, how it chases prey, the type of prey it needs, for example. And that's the functional type. Can be a variety of other functions too, including components of behavior, including mating behavior, habitat preference, types of predator avoidance behavior. So all of these are part of diversity and important in understanding the mechanisms of internal dynamics and understanding how biodiversity evolves and is able to persist in a community. All right, so it's a fundamental feature of communities, species richness and diversity. Fundamentally important, so it's one of the most important things to concentrate on and understand. So richness is simply the number of species. That's just the number, just the count. And by species, we can mean, like I said, taxonomic species or functional types. Diversity is a more complicated measure that tries to incorporate both the number of species and their relative abundance. And the reason for this is that sometimes it's misleading to equate biodiversity with just the number of species. And it's because you could have a community that contains just some substantial number of species, but all but one or a few of those is very rare. So for example, if we have two communities, each having a species richness of 20, namely each has 20 species, but in community one, 19 of those species are so rare that you hardly ever see them, and one is abundant. And in community two, all 20 are equally abundant, then of course we want to say that community two has a higher diversity, even though both have the same species richness. So we need to take into account the relative abundance of each species in order to get a better measure of species diversity. And to do so, we need to recognize the concept of evenness. And this is a tendency for species abundance to be the same across species. So for two communities with the same species richness, the community with the higher evenness should always have the higher diversity. But what if two communities differ in both richness and evenness? Then how do you account for both? How do you balance both against each other to get an overall measure of diversity? For example, if one species is higher in evenness, but the other species is higher in richness. Well, there are several measures that have been proposed as measures of species diversity that take into account both evenness and richness. Three are the most common encountered in the literature. So let's look at all three. So the first is Simpson's index. And this is just the probability that two random individual organisms are of different species. So to calculate this, consider that this probability is 1 minus the probability that the two random individuals are the same species. And what is the probability that they're the same species? Well, just imagine you put all the individuals, write down their species identification on a piece of paper and put them in a hat and just draw at random. So the probability that the two draws are the same species is the probability that they are both species 1 or both species 2 or both species 3 or both species 4 all added up, right? Which is the probability that they're both species 1 plus the probability that they're both species 2 plus the probability added up over all species. So, okay. But for any species, the probability that two individuals are both that species is just the probability that the first random individual is that species times the probability that the second random individual is that species. This is just the independence product rule, remember? And these two probabilities are the same. Okay, so they're equal to the relative frequency of that species in the community. So the relative frequency is the abundance of that species divided by the total abundance of all individuals. And that's the probability that a random individual belongs to that species. So if the relative abundance of species A is 0.1 or 1/10, and the probability that a random individual of species A is 1/10. And the probability of two random individuals of species A is 1/10 times 1/10, which is 1 in 100. Okay, so here's two communities. They both have two species, but community 1 is 99% species A, and community 2 is an even community, equal abundance of the two species. So let's calculate these probabilities. So what's the probability that two individuals are the same species in community 1? Well, it's probability that the two random draws are species A, which is a product of the two relative abundances of species A, plus the probability that the two random draws are species B, which is this product here, and that's 0.98. So that's the probability that two individuals are the same species in community 1 and just a random draw. And the same calculation in community 2, it's 0.5. All right, so how do we convert this to diversity? Just take 1 minus this probability. That's the probability that two random individuals are different species. Okay, so this is the probability that the same species, right here. So 1 minus that is the diversity. So the probability that the two random draws are the same species in community 1 is very small. It's 0.02. But in community 2, it's 0.5. So 0.5 has the higher diversity. So in general, if you have a large number of species in the community, you just take each relative of abundance and square it. If we add up all these square relative abundances for each species, then we'll get the probability that two random individuals are the same species. So now if we take 1 minus this probability, we get the probability that two random individuals are a different species. So this probability is called Simpson's diversity index. That's the top one here. That's fairly commonly used. So it makes sense as a diversity index. For any community, we want the diversity to be higher if the probability that two random individuals are different is higher. However, there are criticisms of Simpson's D. Generally, it is better to have an index that is analogous to richness. In other words, sometimes we want to have a kind of richness, namely a kind of number of species, and sort of an effective number of species. So, for example, if we have a community where the Simpsons index is 0.5, that means the probability is 50% that two random individuals are different species. So maybe a better index than 0.5 would be a number of species, a kind of richness. So one way of converting this to a kind of richness is to ask, if we had a community in which all species were the same abundance, then how many species would we need to have in the community in order to get a D as 0.5? Or a probability of two random individuals being different species equal to 50%. This community would have two species. So this is another index of diversity, namely Simpson's inverse index. That's the second one here. So Simpson's reverse index, I, is just 1 over 1 minus D. This is the species richness of a community in which all species are equally abundant and for which the probability that two random individuals are different species is D. Okay? So this index depends only on D. It doesn't matter how many species are in the community. So if there are 1,000 species in the community and the probability that two random individuals are different species is 50%, then I equals 2, basically. So if there are 10 species in the community and the probability that two individuals are different species is 50%, then I equals 2 again. So this is sort of the effective species in the community, because in either case, there's only 2 effective species there. So Simpson's inverse index I gives a number of effective species in the community, taking into account the relative abundance of each species, no matter how many there are in reality. So in other words, no matter what the actual species richness is. So there are many indices like Simpson's inverse index that calculates the effective number of species in the community. And these all discount rare species by some different amount. So depending on how much you want to discount rare species, you'll get slightly different numbers of effective species. That's basically the idea here. And there's no consensus about which is best, what the best way to discount rare species is. So most people just use Simpson's index or Simpson's reverse index as, they're the simplest. Probably the inverse index is the simplest to understand. And the third index that's commonly used is the Shannon-Wiener index, H, which is minus the sum of P times the log P, whereas P is just the relative abundance of species I. So you add these up for all the species. So the Shannon-Wiener index is based on information theory. And it shows us how much information is contained in the relative abundance numbers of each species in the community. It turns out that the Simpson's inverse index I and H are related. So H is approximately equal to the logarithm of I, or I is e to the h power. So if you take e to the h power, that gives us the effective number of species in the community. Another estimate of that using the Shannon-Wiener discounting of rare species, which is very similar to Simpson I. So one of these three methods will give us a measure of the level of species diversity in a community that takes into account both the number of species and the relative abundance by discounting the importance of rare species by some amount. So if you look at a study on biodiversity, you'll see one of these three, definitely. Or sometimes you'll see all three, because many studies report the results of all three diversity measures, since there's no consensus among researchers as to which is best. Plus, all studies will report actual species richness, just the number of species added up. All right, so we should also talk a little bit about evenness and how to calculate evenness. So here we're using the Simpsons. If we're using the Simpsons inverse index, I, then the effective number of species, the I index, is 1 over 1 minus D, okay? And so that would be the number of species if they were all maximum evenness, okay? And all the same relative abundance. That would be 1 over 1 minus D. So if the actual number of species, if you just count them all up, turns out to be the same as this, then that would mean that all species have the same abundance and evenness is maximum. Otherwise, the total number of species in the community is going to be greater than this. So what would be a good measure of evenness? Well, evenness could be measured as simply the number by which you multiply S by in order so that that product equals the effective number of species. So S is always going to be greater than the effective number of species, I. So whatever number you have to multiply S by to get this, then that's evenness. So evenness is going to be 1. In this case, evenness would be 1 right here. If the number of species is equal to the Simpson's inverse index, then evenness would be 1. Otherwise, evenness is going to be less than 1 here, so that when you multiply it by S, you get this number here. So species evenness then would be 1 over 1 minus D times S. And S is just the total number of species that you see in the community. Just count them up. Don't discount them in any way according to their abundance. Okay, so how do you measure though? I mean, if you think about it, just counting up the number of species in the community is not a simple proposition. What is even meant by the number of species in the community? That S that we were looking at. Well, communities are defined as a local assembly of species at a particular location together in the same place at the same time, and therefore capable of interacting. Within any location, however, species are not distributed at random. There will always be some heterogeneity. If you think of the shallow water along some shoreline, a cove bounded by 2 headlands, and we're interested in the fish community, then you will actually find several different assemblages of fish depending on the type of benthic habitat. So seagrass, bare sand, bare rock, rock covered with algae. So although on average there will be different fish species at the different benthic sites, fish are highly mobile and there's nothing preventing them from moving between the habitats occasionally. So if you watch one species long enough, then eventually you will see it spending time in all habitats. Eventually, even if it spends 99% of its time in one of the habitats, if you watch long enough, it's going to be wandering around in all of them, most likely. So then, should the species richness of all these habitats be the same? That doesn't seem right. The argument for saying that they all have the same species richness is that eventually all the species will be at all these habitats along the shoreline. So why not? Shouldn't they all have the same richness? But the argument against species richness being the same at all of these habitats is that obviously if you just sample at any one time and count the number of species present at any one time, it will be consistently different between the different habitats. For example, there usually be more species present at any one time in the rock habitat than in the sand habitat. So shouldn't we report a higher richness at the rock habitat? Well, there's no simple answer to this question. Both measures of richness are valuable and useful, and you might want to report both for the purposes of completeness. So it's valuable to know the probable differences in richness among habitats sampled at any one time and place. It's also valuable to know the number and identity of species that use the habitat rarely and are unlikely to be seen during a single census on a single day. If you do report richness, though, it's important to be specific about what you're actually reporting. Simply saying that N number of species were observed at the site is not very informative. The reader needs to know the sampling and analysis method used to make sense out of that number. Sometimes all that's necessary is to report the number of species observed during a simple short sampling survey because you want to understand how richness differs on average between sites or between types of sites. You're mainly interested in the differences on average. And you don't need to know the total absolute number of species likely to visit the site over the long term. But that number that you report is for richness is meaningless unless you also report the method in detail. Small differences in the method can give you big differences in richness estimates. And comparisons aren't meaningful unless the method is used identically in all the comparisons. All right, one way of estimating the total number of species in a community, including the species that are likely to have been there during your sampling but which you did not see, is to use what's called a species accumulation curve. So the idea behind a species accumulation curve is that there are species in a community that you happen to miss during your sampling. So if you sample again, there's a chance that you'll see some of these species that you missed. And if you sample a third time, you might see a few species that you missed the first two times. And if you sample a fourth time, you might see a few species that you missed the first three times, and so on. So you can plot the number of total species that you've seen against the samples from first to last. Okay. And as new species are added with each sample, the total goes up a little bit. But as you keep sampling, the amount by which the total number of species increase per sample is smaller and smaller because there's only a finite number of species in the community, and the number of leftover species that you haven't seen is getting smaller and smaller the more time you spend out there. So when you plot this curve, it's the shape of a saturating curve. It eventually reaches a plateau and no longer increases, as you have by that time managed to see all the species. So this curve is called a species accumulation curve and is standard in the community ecology literature. Species accumulation curves, or SACs, are usually necessary because you can't keep resampling and resampling until you've seen all the species. That might take months. So generally, you resample until you start seeing the shape of the curve, and that's usually enough. Once you have enough points to shape the curve, then you can statistically fit a saturating curve to your points and then see where the fitted curve levels off. And where it levels off is your estimated total species richness for the community. Okay, in R you can fit a species accumulation curve using the specaccum function in the Vegan package. It's very easy to use and it's very nice. It gives a nice plot too, like this. But note, however, that species accumulation curves apply to the species present just during your sampling period. If there's an annual variation or seasonal variation that you don't take into account, you'll miss all those species coming in other seasons or in then next coming years. So in theory, if you keep sampling over time like this, the curve will continue to rise from all those very rare instances when some extremely rare species just happens to migrate in from, 1,000 kilometers distant, until all species in the total species pool in the region have migrated in. So it might be true that in an absolute sense, if you wait long enough, the total potential species richness at any given site is just the number of species in the total regional species pool, because they'll all show up there at some point. But of course, this is not going to be useful information. Generally, in any study, you want to make practical decisions or test hypotheses regarding the average number of likely to be encountered at any time for conservation purposes, or to understand the immediate mechanisms operating to determine the number of species present at one time rather than the total number likely to exist there over a long term. So the purpose of the species accumulation curve is to get a better idea how many species are likely to be present at the same time within a particular sampling area during a particular sampling period by making a reasonable inference about how many were there, but maybe you were not able to see during sampling simply because you can't sample and observe everything, and see everything with the effort needed to see all of them including the rare ones. All right, but this is a standard method, and the standard implementation of it people use is the function in R. And so what you generally do is, instead of just comparing the number of species, you compare the results of the fitting the species accumulation curve. So you could compare the endpoints of the species accumulation curve, okay, and use that as your estimate of the species richness at any location or with any particular habitat. All right, one of the most fundamental questions asked by community ecologists is the fundamental question of biodiversity, which is essential for conservation and management over the larger landscape scale. And so, namely, how is species richness and diversity partitioned across the landscape? And to answer this question, usually some measure of effective richness or effective number of species is measured and analyzed. And this effective number is most commonly Simpson's inverse, I, measure at the local scale, the effective number of species. If you discount rare species, as we have seen, because rare species could be problematic. So when we talk about types of richness, okay, we're going to use the term richness, which means not literally the number of species, right, but rather the effective number of species with the rare ones discounted, as calculated, for example, by Simpson's I for a given site. So people also use the term diversity, as in, for example, biodiversity, to essentially mean the same thing, the effective number of species with the rare ones discounted. Or alternatively, the probability that two random individuals that you draw out of a hat are different species, which is another way of expressing the same thing. Okay? Just another way, another convention. Okay, so you could call this richness diversity, or you could call it richness, and I'm just calling it richness now. Okay, so just keep that in mind. We're talking about richness calculated in a way that discounts the rare species. So basically, we can call it richness, or we can call it Simpson's diversity. All right? But let's just call it richness, just for the sake of simplicity. We're going to call it richness. Effective number of species, not the actual number of species, but the effective number of species with the rare species discounted, not counted the same as the more common species. All right, so there are three types of richness, or you could call these three types of diversity, but we're calling them richness. So the first is alpha richness. This is the species richness at an individual site or habitat. And that would be the Simpson's I, for example. So you calculate this by simply doing a survey or census where you get the relative abundance of all the species observed during your census, and then calculate Simpson's I, or you can use one of the other measures, Shannon-Wiener H, or the e to the h power. So you can do a species accumulation curve also, but the rare species will be discounted in I, so it will not contribute much to the effective number of species. Gamma richness is the total species richness across all sites or habitats. So this is just calculated just like alpha richness, but you pool all your data together and treat it like one site, and then calculate richness the same as before using the same measure, Simpson's I, for instance. Beta richness, this is just the number you multiply by alpha to get to equal gamma. This is the original definition of beta richness. Okay, so there have been later definitions. The most common is that beta is the number you get number you add to alpha to get gamma. So you can either define beta richness as the number you multiply by alpha or the number you add to alpha in order to get gamma. So those are basically the two ways. And the original was the number you multiply it by to get gamma. So then beta is either equal to gamma over alpha, so beta times alpha equals gamma, or beta is equal to gamma minus alpha, so beta plus alpha is gamma. So those are the two measures. So the idea of beta richness is that sometimes you see the same species everywhere you sample because conditions and resources don't vary much across space. The landscape is homogeneous. So in this case, alpha richness basically is the same as gamma richness. You see the same species at every site you sample. So the total number of species across all sites is the same as the number within each site. And at the other extreme, sometimes there is a completely different assemblage of species at every site. There's no overlap. And in that case, the gamma richness is much greater than the alpha richness, maybe is roughly equal to the alpha richness times the number of sites you sampled. So this is true if there's a lot of spatial heterogeneity. So like in the tropics, such that every sampling site is essentially a different site or a different habitat even. So these two extremes are almost never observed. Maybe, at the North Pole, close to the North Pole, close to the, in a tropical rainforest, at the other extreme. But rather you see something in between. So for example, there might be roughly 5 major habitats across some area with species mostly different in the different habitats. Then within a site, you'll see nearly all species present within that particular habitat. And across all types, you'll see the total number of species in all habitats. So if each habitat contains roughly the same number of species, then beta richness will be roughly 5. You multiply alpha richness by 5 to get the total richness across all habitats, because all habitats contain all the species, basically, from the 5 habitats. So beta richness is a nice indicator of the degree of spatial heterogeneity of communities, how different they are across space. So if you define beta as the ratio of gamma to alpha, then it's roughly the effective number of different habitats over your sampling area as perceived by your community. And you may perceive it quite differently. So the beta richness in English is also called the species turnover, which means the amount by which species are lost and replaced by new species in communities as you move from site to site. And keep in mind, 2 terms are used. Many people call beta richness beta diversity, but it means exactly the same thing. I'm using richness because it's easier to think about as a count of the number of species rather than an abstract idea related to information theory or probability. But keep in mind that R.H. Whitaker's original definition of the idea, he called it beta diversity and not beta richness. There does seem to be a trend away from using the term diversity in the literature, which can be ambiguous. So one final note on the concept of beta diversity, the idea has expanded into several other more complicated measures, one of which is simply the overall distance or difference between 2 communities, such as the Bray-Curtis distance. This is also a kind of turnover as it measures the extent to which species are lost or become rare and are replaced with different species when you compare 2 communities. So at the moment, the idea of beta diversity is a bit of a mess in the literature with maybe over a dozen different competing versions of the idea. Everybody's promoting their own preferred measure, which is all the more reason to use the term beta richness when focusing on a count of the number of species, a more concrete idea. Also, probably a good idea to just use distance between 2 communities rather than beta diversity when you're referring to the Bray-Curtis or some other measure of distance or difference or dissimilarity, and not to confuse all these things. But, the idea of beta richness is a good one. It's the extent to which communities change in units of effective species across a spatial dimension or in time during succession. And there's a huge and increasing number of field studies of beta richness in all kinds of communities, including marine communities. And these are basically what are popularly called biodiversity research, okay? If beta richness is high, then something is causing a high species richness across a spatial dimension. And it's often a diversity of natural habitats, which is revealed through follow-up research, including experimental manipulations. And these experiments inform us of the environmental factors maintaining that diversity and that are important focus of conservation and management activities. Okay, here's some representative examples of marine biodiversity studies. Okay, so this example of Rivandinera, I guess, is how you pronounce his name, 2002. Looked at latitudinal trends in species richness and diversity, found basically that the regional total species pool, indicated by their species accumulation curves, had the highest species richness at an intermediate subtropical latitude around 30 degrees, right here. But that there was no latitudinal trend in species richness at small-scale sampling sites. Just average species richness, without looking at the accumulation curve. And this indicates that different processes might be occurring at local and regional scales. Or it could be that this is just a lot of noise and we're not seeing anything here. He just draw this graph suggesting that we have this maximum beta diversity at a latitude of about subtropics. And this is in rocky intertidal communities. So the lack of any trend by latitude they found in the small-scale sampling sites, they interpreted as possibly be caused by competition. So competition might be an important factor determining species richness at local sampling sites. So it might be that as a result of competition, that's a process that keeps the number of species at some constant low level at any local site. But they found that beta diversity, and they use the Jacquard index, which is similar to the Bray-Curtis. And again, there's not a strong consensus about what dissimilarity index is the best to use. So you can calculate the Jacquard just as easily as the Bray-Curtis in the R-Vagan package. So they found the highest beta diversities at around 30 and then again around 20, but it's really hard to know if this is real or just noise. And they interpret this as implying that there are relatively abrupt changes in communities at those points, at 20 degrees latitude and at 30 degrees latitude. So that we're going from one oceanographic regime here to another oceanographic regime here, and there's another regime here. So it's basically what they think might be going on. So that there's regimes of conditions and resources that change at these points and cause changes in the community. But this is still somewhat speculative and more research is required to know what the changes are at those latitudes, at least in the rocky intertidal habitat that they were looking at. All right, Richard Brodeur et al., colleagues, did a nice example of an ordination study of fish communities, this one off Washington and Oregon states, in the U.S. And this is basically a government study, the Northwest Fisheries Science Center. Okay, what they did was they went on several transects with a boat with a trawl net, midwater trawl net, and made fish trawls at all these little plus signs from the Push Washington down to Cape Perpetua. And these are all the species they caught in their nets and the relative amounts, the mean number of fish per kilometer that they towed the net. On all of these dates. And this is the percent frequency of occurrence of the 27 most frequent species and life history stages, young and adult. So these are the different cruises. And these are the numbers. So, for example, northern anchovy was found in 25% of all the cruises, 25% of the 40 collections. All right, and here's a little summary of the biodiversity right here. We've got the Shannon and the Simpson. And the Simpson here, the diversity measure, is basically the probability that two random individuals were separate species. Notice they're doing the probability Simpson's measure, the D measure, rather than the I measure. And they also calculated the evenness of the fish community. This is the number of taxa, species or genera, that they found in the June 98 cruise. And here's their ordination plot. And they did, notice, an NMS plot here. And these are the two axes that they found that allow us to view each of these clusters, each of these sampling points, and they're color-coded by cluster on a map, on a two-dimensional map. So this ordination shows that the most important factors controlling the fish community were nutrients along the x-axis and latitude along the y-axis. You see that latitude is almost perfectly correlated with the y-axis here, and nitrate increases towards the right here, so the nitrate concentration increases towards the left, and the sea temperature increases towards the right, and the sea surface temperature increases towards the left, the distance from land towards the left, the depth increases towards the left, and the water transparency increases towards the left. So land east and the mainland would be on the right, north is to the top, south to the bottom, and west is towards the left. And we're going deeper and deeper water, more and more oligotrophic, more transparent, farther distance from shore, higher temperature. All right, so this is a coastal upwelling area. So upwelling is going to occur close to land. So towards the land, we're going to have increase in nitrate concentration. And as you go farther away from land, you have fewer and fewer nutrients in this direction. So low nutrients are correlated with higher sea surface temperature, greater distance from land, deeper ocean water, and more transparent water. So high nutrients to the right, low nutrients to the left, north to the top, and south to the bottom. So all these things are negatively correlated with upwelling. Nitrate is positively correlated with upwelling. All right, and we can also plot the fish species, or at least the most common ones, on the same ordination axis as before. So the fish species to the right are associated positively with upwelling areas, and those to the left with downwelling areas. Those up with more northern and down with more southern transects. So note that most species are positively correlated with upwelling, concentrated to the right of the ordination diagram. So you could say this is sort of the upwelling community of fish, and this is the downwelling community of fish. We've got mackerel, blue shark, ocean sunfish, Pacific saury, all associated with downwelling, low nutrient situation. And then we have more northern species, the jack mackerel, up to the top, and then more southern species, ironically the northern anchovy, farther to the bottom of the ordination diagram. And interestingly, they also did a dendrogram for the species. So we can see basically these clusters here. They basically proposed as different fish communities in different areas. So here we have more southern species. Then we have the jack mackerel to the north, and in the more nutrient poor areas, then these fish in here tend to be more highly associated with the upwelling. So this would be part of the upwelling community right here. And interestingly, this table of species diversity and evenness values shows that there was a drop in diversity right here in June 2000 when there was an overabundance of one species, anchovy, that year after a strong La Nina weather event, which was high upwelling, colder water, higher nutrients. So this shows the effect that normal annual variation can have on environmental stochasticity, which can cause a large change in a community property, namely the alpha richness in this case. So Carrie Ellingson studied soft sediment macrobenthos community structure, found that beta diversity increased in communities with species having restricted geographic ranges or across different depths or across communities with different sediment grain sizes. So species accumulation curves showed that the total maximum species richness was much larger than was possible to sample in individual samples. So this is the cumulative number of species, just the raw richness of polychaetes, mollusks, crustaceans, and echinoderms. So see that the largest number of species most likely are going to be polychaete worms. And here we have just richness, or I guess we can call it diversity measures within single samples. So we've got the S measure, which is just the species richness. It's a count of the number of species. And then we have the e to the h power. That's exponent of the Shannon index, which should be close to the Simpson's inverse index. Okay, so they should be very similar to each other. So we've got the species richness, the raw species richness, e to the h power of the Shannon index, and then the inverse Simpson index of diversity for these different sample numbers for polychaetes, crustaceans, mollusks, and echinoderms. So you can see from these samples that the number of species, the effective number of species, was just in the tens, maybe 80 or so max for the total number of species, and the effective number was about half or a third that. But then if you look at the species accumulation curves, you got numbers up into the hundreds for polychaetes and above 50 for, I guess these are mollusks and crustaceans. So those are much larger numbers total than what you're seeing in the individual samples, which is a common thing to observe, which is a little bit problematic, okay, but something that you need to take into account. All right, and then so she saw an increase in beta diversity as the area increased, the area that you consider increases, which indicates that the average difference between two sampling sites increases, between two random sampling sites increases as the area increases, which means that this is a habitat effect, the habitat heterogeneity effect. So the bigger the area that you consider, the more likely it is that there are different habitats included in that area. So the differences among the samples start to increase there. So this would be a basic effect. The slope of this curve is a basic indication of the amount of habitat heterogeneity in the area that's being sampled. Here's a plot of the depth difference between sites. So every pairwise comparison of two sites, and then you look at the difference in depth between those two sites and also calculate the Bray-Curtis similarity between the two sites, which this indicates that as the two sites are more separated in depth, the more dissimilar they become. The dissimilarity goes down. And this just shows that the importance that depth has on driving the community composition, the species that are present. So you see different species or different communities at different depths. And again, the slope of this line is the effect of depth, the effect that depth has on causing differences in the community assemblage for these samples. Okay, another subject, an important subject, which is the effect of predation on the community. And so this is an interaction constraint on the community. So predation and the role of predators in the community, the type of behavior they use and the type of predation they use, can have an enormous effect on all the species, essentially all the species in the community. And this is a common phenomenon that has been observed in many communities, important and near ubiquitous in nature. The predators have an effect, especially top predators, on community structure, especially on alpha richness in the community. Okay, and there are two major types of predatory impacts on community structure that have been well studied, and I just illustrated the simple diagrams right here, the food web diagrams. And these are the trophic cascade, the TC effect, and the keystone predation effect, which is the KP effect. In the trophic cascade, a top predator feeds on a consumer resource species, or a compartment just below it in the trophic chain. Therefore, the predator has a negative effect, a direct effect on the fitness and population size of that prey. However, the prey also feeds on a consumer resource below it on the trophic chain, and therefore the top predator has a positive effect on the fitness of this species 2 levels down. And in this diagram, we're assuming that the predator does not feed on this B species 2 levels down. So a trophic cascade is just a positive effect here of the predator 2 levels down at the very least. And of course, if there's an additional positive effect 4 levels down. So an influential top predator with a high consumption rate, if it causes a trophic cascade, causes negative, positive, negative, positive, alternating effects on the trophic levels beneath it. So it's a minus, plus, minus, plus, minus, plus going down the cascade, always alternating negative and positive effects. So this alternation of negative and positive effects down at the food chain, it's called the trophic cascade, like water cascading down a waterfall. So the species the top predator preys on, the species that it preys on right here, is called a mesopredator. Meso because it's in the middle, it's not the bottom or the top. So when the top predator is taken out of the community, which often happens in cases of overfishing because overfishing often targets these big top predators, then we say the mesopredators are released from predation, their populations become more abundant, and their predation rates on prey species increase and might drive the prey population down. So trophic cascades is also the consequence of the elimination of a top predator, which is happening everywhere in every ocean of the world, which causes a positive, negative effect, at least in principle, down the food chain. And of course, this is happening in the Mediterranean and the Adriatic. Pretty much every fish community we're looking at has has an absence of these top predators or extreme scarcity of these top predators, and the mesopredators are overabundant and have driven down the populations of their prey species. The second major form of predation control impact on community structure is called keystone predation. And this is when a predator preys on a common dominant competitor, thereby lowering its population density below its shutout level, preventing it from shutting out its competitor. If this prey is a dominant competitor that excludes many other species, then keystone predation prevents this exclusion and protects all these other species in the population and therefore maintains diversity in the community. So the predator preys on this dominant competitor, has a negative effect on that, and this dominant competitor has a negative effect on many other species that it competes against and eliminates them. And so the predator actually has an indirect positive effect on all these species that lose out and are excluded as a result of competition with this dominant competitor. Major example of trophic cascades is the one observed by Frank et al. Ken Frank, in the cod-dominated ecosystem in which large benthic predators were overfished and driven to very low levels. So remember, in top-down trophic cascades, if you eliminate the top predator, that's going to have a positive effect on the mesopredator just below it, which is going to result in a negative effect on the zooplankton that are fed on by the forage fish, the mesopredator, and then a positive effect on the phytoplankton below that. So here we have a time series here of cod biomass is the dashed line. So in 1980 was quite high, and then it has plunged down to very low levels over the next 20 years. And here we have a time series of all groundfish landings and biomass, and along with the seal biomass. And we can see from 1980 on, there was a collapse of the groundfish biomass. All the benthic predator guild here collapsed, and then the landings went down. The fishery landings went down in parallel with the loss of the biomass. So what was the response to the lower trophic levels? Well, the prey of these benthic fish increased. These are the small pelagic nekton, shrimp, snow crabs, which greatly increased from around 1990 on. So these are the mesopredators, which were released from predation by the large benthic predators lost from the cod ecosystem. And what happened to the prey of the mesopredators, namely the zooplankton? Well, it decreased over the same timeline. From this mean here down to this mean over here, so not quite half. So it decreased over the same time that the mesopredators increased. And what happened to the phytoplankton prey of the zooplankton? Well, it increased over time, correlated to the decrease in the zooplankton that was feeding on it. So this is a perfect example of a trophic cascades and this cod groundfish ecosystem caused by overfishing of the top predators in this system. And a subsidiary effect of the loss of the benthic fish was the release from competition of the gray seal, which competes with the benthic fish for the same food, the small pelagic fish and benthic invertebrates. So the gray seal benefited from the loss of the bottom fish and the increase in their own food supply as a result. So, classic example of a trophic cascades affecting 3 trophic levels in alternating directions. As predicted, increase in the Mesopredators, decrease in the zooplankton, increase in the phytoplankton, just as predicted from this top-down trophic cascades model. So another example of trophic cascades in the Black Sea is Daskalov and colleagues' study of 4 trophic levels, just like the last one with the cod. Overfishing depleted the top predatory fish, which resulted in an increase in small fish, their prey species, which resulted in a decrease in zooplankton and increase in phytoplankton. And on top of this trophic cascades, as we've seen, there's a second shift caused by the introduction of the invasive jelly, Nemiopsis ladii. So the jellies increased, and this was facilitated by human cultural eutrophication, which additionally caused an increase in phytoplankton and ultimately a loss of oxygen in the deeper zones of the sea. All right, so those are classic examples of trophic cascades, marine examples. So let's look at the classic example of keystone predation, also a marine example. So the original example that gave this dynamic a name was in the rocky intertidal habitat, this time in Washington State in the US. And it's also an example of a removal experiment, one of the first in the rocky intertidal in which a species was removed, namely the keystone predator. And the researcher monitored the community's response to the predator removal. And it was Bob Payne's PhD thesis, basically. So the response to the removal was a sharp decrease in the total alpha richness of the community because the predator preferentially consumed the dominant competitor. And when the predator was removed, the competitor increased to its shutout level and excluded a large number of competitors from the community. So this experiment made Robert Payne famous at the University of Washington in Seattle. So he noticed there was a sharp boundary in the mussel beds. Right here is a picture from his original paper. So above the boundary, mussels were abundant. This is Amyllis californianus. And there were a few other species attached directly to the rocks. There are species attached to the mussels, but few other animals occupying primary space attached to the rocks wherever the mussels were, because they outcompeted everything. And below the mussels, another shot, below the sharp boundary, mussels were almost completely absent, and instead of the mussels, they were replaced by many species of barnacles, snails, other invertebrates, marine algae attached to the rocks. So he wondered what was causing this sharp boundary. And what he observed was that the sea star, genus Pisaster, a predator of the mussels, came up into that lower area below the mussels where there were no mussels regularly. So he hypothesized that predation on the mussels by the sea star, the ochre sea star Pisaster ochraceus, the same species that was decimated recently by the wasting virus disease. This sea star was eliminating the mussel from the lower areas in the intertidal. So it's a pretty obvious hypothesis to come up with, when you go down at low tide and you see something like this. So to test this hypothesis, he simply removed the sea star from the rocks, went down at low tide, just walked down into that area, grabbed the sea stars, removed it from the rocks, tossed it into the water over an area of around 50 meters of shoreline, and came back many, many times to do the same at low tide to keep them off the rocks. So this is Bob Payne many years later, a more recent photo. Come back to the same area. Posing for an interview. So over the period of a few years, the mussels gradually moved down the rocks to lower vertical elevation, eventually almost a couple meters lower. And this is table 6 from his paper. This is the number of Pisiaster removed, so we counted them all up. And from the south site, this is the distance that the mussel moved down. After the Pisiaster was removed, tossed off the rocks into the water. And this is the movement compared to the control site. All right, as the mussels moved down, they competitively excluded all other species attached to the rock, invertebrates and algae, mainly barnacles, snails, and macroalgae. And they did this by overgrowing, smothering, or by crushing other species between the shells of adjacent mussels. So in the presence of the sea star, found a higher alpha richness on the rock, including barnacles, macroalgae, limpets, snails, anemones, urchins, and sponges, all attached to the rock surface. Okay, so all these species were attached to the rock surface where the Pisiaster had been grazing, but when you take away Pisiaster, all these species are outcompeted and excluded by the mussel. So in the absence of the sea star, we had eventually almost none of these. They were competitively excluded, killed by the shells of the mussel, aggressive competitor. So the sea star preys on the mussel, and the mussel competes with many other species for rock space. And a large number of species, around 15 obvious species. And when sea stars are present, they move up into this area when the tide comes in and consume the mussels. They just pull them off the rocks with their tube feet and then pull open the two shell valves with their tube feet and then evert their stomach into the between the two shells of the mussel and just digest the soft tissues inside the mussel with their stomach. And this is how they feed on those mussels. All right, but then when you remove the sea stars from this area of diverse species below the mussel bed, below the sharp boundary, over time, the mussels gradually spread down into this area and outcompete and remove all of those diverse species until they're gone from that area below the mussels. So there's an elimination of roughly 10 to 15 species. Right. Payne named the sea star a keystone predator, meaning that it had an importance in the community far more than its abundance. Like a keystone has for a building, even though it's just one stone. So if you look at a particular species any particular species. It might have a low biomass or a high biomass in the community, and it might have a low total impact on the community, and it might have a high total impact on the community. So a keystone species is a species that has a fairly low biomass, but a very high impact on the community. A dominant species, on the other hand, has also a great impact on the community because that species is dominant and also abundant, high biomass. And an example of a dominant species is the California mussel. It's a very high biomass, and it is the dominant competitor in the rocky intertidal community. So a keystone species also has a high impact, but it's not abundant. It has an impact far beyond what you'd predict from its biomass or abundance in the community. So most species that have a low biomass but a very large impact on the community, are predators whose consumption of other predators or competitors is what causes the impacts on the community. Another example of a keystone species is the sea otter in the marine environment, proposed as a keystone in kelp beds of the Northeast Pacific. So the otters prey on sea urchins, among other things, which in turn prey on the kelp and most other macroalgae, and as we know, can cause urchin barrens. And recently, the sea otters have declined in abundance in Alaska for unknown reasons, possibly due to increased predation by killer whales. And areas where sea otters have declined have experienced almost complete loss of kelp beds. We have urchin barrens where the sea otters have declined. Loss of kelp beds and many other associated species to the kelp beds. Obviously, it's a substantial, huge loss of alpha richness across the kelp bed community. This apparently was caused by atrophic cascades in which the loss of the sea otter allowed explosion in population of sea urchins, causing loss of kelp beds. And once the kelp beds were lost, then all of their associated species were lost also. Abalone, smaller invertebrates, crabs, many sessile invertebrates, smaller predatory fishes and invertebrates, and larger fishes and octopus that preyed on them. So such a catastrophic loss of alpha richness or biodiversity, as the media reported, simply due to loss of this one keystone predator here, the sea otter, in these communities. And you can read about this in this original paper by Estes et al., 2004, a review of all of these processes. So this is urchin biomass before the decline, after the decline of the sea otters. Urchin biomass increasing at 2 locations in Alaska. Grazing intensity on the kelp increasing as a result. Kelp density plummeting as a result. Decline of otter populations from 1972 to 1997, several locations. Surveys of the shoreline of sea otter density. And whales harvested. So what do whales have to do with this? So large whales were harvested over the same period that the sea otter declined. These are large whales that were normally preyed on, the sick or the older individuals, by killer whales. And once these large whales were gone, killer whales may have shifted to other prey, like other marine mammals. such as a sea otter. That might have been the cause of the sea otter decline, resulting in the collapse of the kelp ecosystems. This is an example again where a fairly uncommon species exerts a disproportionately large effect on the total community, therefore keystone species. And the keystone effect in this case is through primarily trophic cascades. Predator apparently has a positive effect on a large number of species underneath the main mesopredators, the urchins. There are many examples of the release of urchin populations with the depletion of their predators, causing collapse of algal communities and their associated species in many places worldwide. So, for example, in New Zealand, Shears and Babcock paper here shows that in marine reserves, where urchin predators are more abundant, spiny lobsters and fish in the Sparus genus, they reduce the urchin population, which allows macroalgal beds to persist. So here's study sites in New Zealand. And here are the results. Outside the reserves, the predators are rare and urchin barrens are more common. So these hatched areas are the urchin barrens, and these are non-reserve areas. And then within the reserve areas, we have smaller spatial coverage of these urchin barrens, and more abundant fucoid algae. And we've got the kelp beds at lower depth in both areas. Same thing happening in the Mediterranean. Guidetti and Sala here. In this case, fish in the sea bream, Diplotis genus, consume the urchin Paracentrotus, which consumes the macroalga Cystocera. And again, trophic cascades apparently occur and can be demonstrated by observational data, again, inside and outside marine protected areas. So here's the marine reserves and no-take zones that they looked at and their locations. Northern locations in the Italian side of the Adriatic. So here's a plot of fish abundance and biomass inside and outside the reserves, arranged into large piscivores, small piscivores, invertebrate feeders, invertebrate feeders too, detritivores, planktivores, herbivores, all these different fish species right here. And what we have plotted here is the log of the ratio of abundance inside and outside the reserves. So if this number is positive, then there's higher abundance inside the reserve. So you can see the vast majority of species had a significantly higher abundance inside the reserves. 95% confidence intervals here, including the Diplotis predators of the urchins. These two Diplotis species. Significantly higher abundance inside the reserves. And then another diagram here in which these were all just lumped together. These different functional categories of the species, of the fish species, were lumped together, and another analysis was done, showing a significantly higher abundance of invertebrate feeders and biomass inside the reserves. And the effect was significantly increased by age of the reserve. So significant effect in the subordination. So these are all the reserves. So older reserves here, younger reserves here. And this shows as the density of diplodous fish predators of the urchins increases, the number of sea urchins, the density of sea urchins per meter squared goes down. So another example of atrophic cascades in which a keystone predator has a major effect on the community by preventing urchin barrens from occurring through its consumption of the main prey of the urchins. Okay, so just to step back, we're looking at internal dynamics, the internal dynamics constraints on a community. So looking at trophic cascades and keystone predation, sometimes the reason you do see a species in a local community is because a keystone predator is there and doing its job. And the reason a keystone predator is there often is because you're within a marine reserve and it's been protected from excessive harvest. And sometimes the reason something is not in the community is because there's a keystone predator that's missing from that community. So next time you see a healthy algal community, when you're snorkeling, the reason might be because there is a top predator or a keystone predator there whose influence ultimately has an effect on the algal community, on the herbaceous community at the base of the food web. Another kind of interaction dynamics, which has a large effect on who is in a local community or not, are bottom-up effects. So we've been talking about top-down effects, namely control of community assembly and organization by the consumption of prey by predators. So predators are at the top of the food web, and their cascading effects they have on the rest of the community below them in the food chain. The opposite of this is bottom-up effects, namely effects on the abundance of species and the diversity caused by changes at the base of the food web rather than at the top of the food web. So for example, Summer et al. Pointed out that there are obvious examples of bottom-up effects, and we've been talking about these bottom-up effects already. And these are the differences between the three major marine trophic systems. We've got upwelling systems, oligotrophic systems, and cultural eutrophication systems. So obviously nutrients at the bottom of the food chain has an enormous effect, or can have an enormous effect, on the entire community. So the Black Sea and the Gulf of Mexico, for example, provide prime examples of the bottom-up effects of the entire community on the entire community as a result of anthropogenic activity. Eutrophication as a result of organic pollution, agricultural fertilizer, and waste runoff and sewage cause collapse of the food chain and massive loss of biodiversity. Increase in nutrients causes blooms of algae, often toxic, and throughout the summer, the thermocline prevents the mixing of top and bottom water, and as a result, hypoxia develops in the bottom and kills a large active predatory nekton, basically a fish die-off. So a fish and grazing-based community is then replaced by a jelly detrital-based community. Jellies invade and become dominant in the nekton, sometimes so dominant it looks like you could walk across a sea on the backs of the jellies. So this paper talks about this interesting idea of a regime shift. So that's the idea that there's more than one stable state for a community. And this idea is illustrated by bottom-up effects. In the Black Sea, there appears to be two stable states with the possibility of switching to one or the other. The two stable states or regimes are the fish-dominated grazing system and the jelly-dominated detrital system caused by eutrophication. And the idea is that once the jellies dominate, then this is a stable state because their predation on fish larvae makes it very difficult for fish to recover, even if the eutrophication is stopped. So the question is, can this be moved back to the original system again once the jellies are abundant? Well, it appears that in nature, this is very difficult to do. Without some sort of human intervention to push it back into this state. So another important example of bottom-up control of communities is the example of coastal upwelling systems. So coastal upwelling systems occur in eastern boundary currents in the ocean, as we've already seen, where winds create a surface current that is deflected away from land by the Coriolis effect as a result of the rotation of the Earth. And that loss of surface water is replaced by deep water, which has low oxygen, is cold, high in nutrients, including silicon. And the upwelling during the summer allows high NPP to continue throughout the summer, where otherwise a thermocline would develop and prevent mixing, leading to low NPP, which happens in areas where there is no upwelling, which is most of the ocean, most of the coastal areas too. And we know the importance of upwelling by observation of what happens when upwelling stops or slows way down. So upwelling can shift to downwelling in these same areas, in the formerly upwelling areas, in eastern boundary currents of the Pacific Ocean during the weather and climate phenomenon known as El Nino, which is a climate weather regime shift that occurs regularly every 3 or 5 years, causing changes in the weather over the entire Pacific Ocean basin, northern and southern hemisphere. It's a major effect. So El Nino is caused by the breakdown of Walker circulation at the equator. So this means that the trade winds, which are normally east to west at the equator, they weaken and stop. Normally the trade winds create a surface current to the west, causing surface water to build up to higher elevation on the western edge of the Pacific at New Guinea. So this is a warm pool of water causing rising air masses and rain thunderstorms on the western margin of the Pacific Basin at New Guinea and neighboring areas. The rising air then completes the cycle by veering back towards the east in the troposphere, and then it descends at the eastern boundary current at South America, creating areas of cooler and drier climate. So on the eastern edge, this descending air creates high pressure system with coastal winds creating coastal upwelling. This system occurs in all the eastern boundary currents in the world. And this whole system, this Walker circulation, for reasons that are unknown, is reversed during El Nino, starting in December and January. So the Walker circulation weakens and reverses, creating the warm pool now at the eastern boundary current and descending air here rather than here. So here's the normal circulation with the normal Walker circulation, and here's during El Nino All of this ascending air and warm mass moves back over towards the other side of the basin. So heavy rains and the high pressure system is replaced with a low pressure cyclonic storm system over here, and the winds are reversed. So as a result, the surface currents reverse direction, and instead of flowing towards the equator, flow away from the equator along shore. And these reverse currents create downwelling and loss of nutrients in these eastern boundary currents. And this regime may persist for months to a year, continuing throughout the winter, spring, summer, and fall. And as a result, the upwelling based system here on the eastern boundary, that system collapses and is replaced by downwelling. And the nutrient poor waters from offshore are pushed to the coast, bringing poor nutrient conditions throughout the coastal zone. And these surface waters downwell at the coast. And as a result, the highly productive waters that are normal in the California and Humboldt currents, Pacific, turn into temporarily oligotrophic conditions. And the NPP collapses to near zero, and the fishery economy collapses. So El Niños happen every 3 to 5 years for reasons unknown. The most pronounced El Niños on record were 1982 to 1983, 97 and 98, and 2014 to 2016. And here's the 82-83 El Nino. A nice summary, Barber and Chavez. There's 53 times more nitrate in normal times, normal years, than during El Nino in various locations. 53 times more, 78 times, 30 times. So we've got a major collapse of nutrient conditions during El Ninos. Chlorophyll A and primary productivity, we've got like 5, 20, 21 times the amounts during normal years as compared to El Nino years. Huge drops in nutrients in NPP during the El Nino event along formerly upwelling waters. Among the animal populations, sardines, jack mackerel, and hake populations and commercial harvest plummeted as the food web collapsed. Notice that shrimp populations increased. This is another physical bottom-up effect. The surface currents switch direction along the coast, which means that any larvae that are mostly passive and dependent on the currents for dispersal are transported in the opposite direction that they usually are transported. So what this means is that species normally transported towards the equator are transported away from the equator towards the poles. So during El Niños, more warm water species migrate with the currents into cooler temperate zones. And it appears that the community experiences a shift towards warmer water species that may persist for a few months after the El Nino. And the total species pool for temperate communities becomes more equatorial and tropical during the El Nino. So some of these species are more tropical species moving in. Janet Hodder and Michael Graybill, two friends of mine, reported on the Oregon coast collapse of the seabird breeding season after the 1982-83 El Nino. So they counted dead seabirds along the beaches and found 2 to 15 times the number of dead chicks along one section of the beach during El Nino than they usually found other years. Then at the breeding season, the next year, there were virtually no chicks fledged and therefore no dead chicks at all along the beach. So during strong El Ninos, the food web collapses, meaning loss of nutrients, very low NPP, very low NSP for several months to over a year, with widespread decreases in populations of many species and loss of diversity in the community. Classic bottom-up control of the community. It's a nice diagram of bottom-up control in a coastal upwelling area. Upwelling brings in the nutrients, which basically powers the entire food web. And so every 3 to 5 years, this process is reduced or completely eliminated, especially during the summer. And we have a reversion of upwelling areas into a system of productivity. Seasonal productivity is more similar to the non-upwelling areas in the temperate zone. Collapse of nutrients and productivity during the entire summer. Okay, so we've seen top-down and bottom-up control of a community. There's a third type of control that's in between the two, and it's a little bit more controversial. It's called wasp waste control, which is probably rarer than the other two. The metaphor is a wasp body with a head as a top trophic level and the tail as a bottom trophic level, and the narrow waist as some mid-level predator that has low biomass. So the narrow waist of the wasp. So this predator has low biomass, but nevertheless controls the populations at both higher and lower trophic levels. You could also call this mesopredator control. The idea is that the mesopredator, abundance of biomass, controls the higher trophic levels through bottom-up effects and also controls the lower levels through top-down cascades. Okay, so wasp waste control is a combination of both top-down and bottom-up control, hypothesized to occur in some systems. All right, the main marine example of wasp waste control is the work of Fauschald et al. In the North Sea ecosystem, which they proposed that the wasp waste basically is herring fish, which can have a bottom-up effect on seabirds and a top-down effect on zooplankton. So here's their diagram showing herring here, and sprat also. So seabirds are subject to bottom-up regulation by their prey pelagic fish, namely herring. As herring increases, the seabirds increase. So that's a bottom-up effect of the herring. And the herring and sprat regulated top-down the populations of their prey, two groups of zooplankton, calanus and krill. So as the herring increases, the krill goes down, its prey. As the sprat increases, the calanus zooplankton goes down, its prey decreases. So the herring and probably other small pelagic fish in the middle of the food chain, in other words, the waste of the trophic system, they propose are the key to understanding and conserving the North Sea marine community and ecosystem. And if the predators of herring and sprat are overfished, then they can increase and have an enormous effect on the entire ecosystem. But wasp waste control is kind of strange because it begs the question, what is regulating the herring and sprat? It can't be regulated bottom up since the herring is negatively correlated with the lower trophic levels. And bottom up control would imply a positive correlation. And it can't be regulated by the higher trophic levels because that would imply a negative correlation, but they observed a positive correlation. Okay, so the way out of this paradox is they're assuming that the herring and sprat are regulated by a top top-down mechanism by other fish that are not here in the diagram. So although they are regulating the seabirds, they themselves are being regulated by a larger predatory fish compartment here in the ecosystem. So they would be regulated in a top-down manner. So you could argue wasp waste control seems to be complementary to both bottom-up and top-down, rather than an alternative. Something has to be regulating the middle trophic level, and most systems probably, most likely, are a combination of both top-down and bottom-up regulation. Okay, final question. Diversity gradients in nature. All right, back to our conceptual diagram. So all of these processes together act to produce the local community. We have dispersal constraints, we have a total species pool, we have environmental constraints, we've got a geographic species pool, a habitat species pool. They combine to create an ecological species pool, and then internal dynamics filters out the species that are capable of coexisting within this community. So the internal dynamics includes trophic cascades, keystone predation, top-down control. Internal dynamics includes bottom-up control through nutrient regulation in the community. Internal dynamics also includes wasp waste control of the abundance and diversity in the community. And one final outcome of this community that is measurable is the number of species in that community, the number of species. All right, in terms of the number of species, one of the biggest questions in community ecology is, why are the tropics more diverse than any other latitude on Earth? What is it about the tropics that results in a larger number of species surviving in the community after all these constraints are acting? So we've got dispersal constraints, environmental constraints, internal dynamics. Nevertheless, all of these processes in the tropics somehow result in a higher diversity than in the temperate zone. So why is this? Is the total species pool higher in the tropics? Are the dispersal constraints less in the tropics? Are there less environmental constraints in the tropics? Are the internal dynamics a little bit more benevolent in the tropics? What's going on? Unfortunately, there is no simple answer to this question, and there are probably many processes going on at the same time. And there's still a lot of debate. So let's take a look at the situation, the science for the marine environment. So this is the general topic of gradients and diversity in nature, and usually the diversity referred to is alpha richness. Okay, but the question also applies to beta and gamma richness, all of which can vary at different latitudes and show higher values in the tropics. So what's special about the tropics, and what does this higher diversity tell us about the struggle for existence and its effect on how communities are organized? Keep in mind that diversity is a bit of a complicated concept, as illustrated by the Rivadeneira study, 2002, that we already looked at. So remember, they found that the species pool, from literature accounts of species ranges, contained the highest number of species around 30 degrees latitude. Okay, right around here, 30 degrees latitude. And the site richness from species accumulation curves at each sampling site was highest at around 30 degrees also, with that perhaps a second peak, maybe around 20, 25 degrees. The sampling unit richness, the number of species with single sampling units, shows lower richness at 30 degrees. Down here, okay? The lowest in the study, right here. But the highest sampling units were at 32 and 26 degrees. So right up in here. So overall, it appears that somewhat different patterns were observed depending on the scale at which you are asking the question. Processes operating within a sampling unit are probably different from those operating at a regional scale. Migration constraints, habitat constraints, internal dynamics, all are occurring at the same time, and all are occurring at different scales, spatial scales that you look at, and they sometimes overlapping. So these are some of the most complex questions in ecology. Sorting these out is the major goal of community ecology, really, and the major goal of biodiversity management. Here's a nice example of such a study, the worldwide diversity of coral reefs. Bellwood and Hughes, 2001. In this study, based purely on published community data worldwide, it was found that the most important factor explaining the alpha richness of both corals and fish at a particular study site is the amount of shallow water habitat. Within about 600 kilometers radius of the study site. So this basically habitat area explained about 40 to 60% of all the variation in species richness across all study sites worldwide, both fishes and corals. Species richness, number of species, biodiversity of both fishes and corals explained by the amount of habitat area, basically shallow water habitat nearby. The species composition at sites differing in shallow water habitat was not significantly different from what you'd expect from just a random sample of migrants, indicating that there is no evidence of any internal dynamics influencing the number of species of either corals or fish. All sites had roughly the same proportion of species from 3 families of corals and 3 families of fish. So the 3 families of fish are the Pomacentridae, the damselfishes, clownfish, Labradae wrasses, and the Serranidae, sea basses, groupers, and others. So no matter how many species were present at a particular site, these 3 families had pretty much the same proportion contribution to the fish diversity, and the same with these 3 most abundant families of corals here. No matter how many species of those corals were present, each of these families had pretty much the same proportion contribution to the diversity of the community. So alpha richness was higher near the equator and lower in the temperate latitudes, but latitude explained a much smaller amount of variation independently of other factors. Latitude right here, only about 10% of the variation alpha richness of both fishes and corals at coral reefs. So this small latitude effect is still unexplained. So most shallow water habitat exists near the equator in the tropics, and this is the main reason the highest coral reef diversity is in the tropics, in the Indo-Pacific right here, sort of the center of it. And there's an additional smaller reason shown by the independent statistical significance of latitude, but this really has only a small effect, and the source of the effect really is not known. Also, there's a significant longitude effect on alpha richness at a site, and this is simply a measure of the distance from the main species pool which is centered right here in the Indo-Australian archipelago, right in here. So the farther away from you are from this, the lower the species richness, and it's a small effect. So the story of coral reef diversity seems to be an island biogeography effect. For the most part, it's a species area effect. So the larger the habitat area, namely shallow water, the larger the number of species we see, both corals and fish at coral reefs. A smaller effect, again, is island biogeography. The distance between the reef and the mainland source of species, namely the Indo-Australian archipelago, also reduces the alpha richness. And another small effect is latitude, with higher alpha richness near, not necessarily on, the equator. And we currently don't know the cause of this effect, or if it's a real effect independent of the others. But it seems to be, as it is statistically independent, and explains about 10% of the variation in the alpha richness. So a nice study by Bellwood and Hughes, nice classic study here. Good example of the kind of study you need to do to understand what is controlling biodiversity within coral reefs, which are a threatened habitat housing a very large biodiversity. And conceptually, it's not a difficult study to perform. You have an alpha richness for each sampling site, and also for each site you have several environmental variables. So to test the effect for each of those environmental variables, you just perform an analysis of variance with the alpha richness as your response variable and the environmental variables as your predictors. And with the ANOVA or similar type of model, some kind of regression, you can see the proportion of variation in alpha richness that is explained by each of the environmental variables. So many studies of this type have been done in the marine environment, asking what the effect of latitude is on alpha richness measured at several scales. What have they shown us about the effect of latitude on marine biodiversity? Well, Helmut Hillebrand, 2004, out of Kiel, Germany, reviewed basically all the 200 such studies up until then in the marine environment and presented the overall results of this paper in MAPS. And he found that there was a strong overall statistical effect of latitude on diversity measured in various ways in the study in marine environments. So diversity measured as alpha or gamma richness is significantly higher near the equator than away from the equator. Another way of saying this is that the correlation between diversity and latitude is negative. So negative here on the y-axis. And it's significant. The 95% confidence intervals don't overlap zero. So this is a significant negative effect for most of these measures. So the effect of latitude was about as strong or stronger in the marine environment as on land in terrestrial environments. And the effect of latitude was stronger in marine habitats than in freshwater habitats, but the effect also existed in freshwater habitats. So the effect of latitude on diversity was almost entirely observed at the regional scale, gamma richness, and was not really a large effect at the local scale, so the alpha richness within the sampling site. And the effect of latitude on the local diversity, basically the alpha richness, was much smaller. Also, the effect of latitude was greater for pelagic open water communities than for benthic or ground communities, and greater for nekton and mobile epifauna than for sessile epifauna and infauna. And the effect of latitude is larger for taxa with a large body mass and for predators at higher trophic levels. And there's no difference in the effect of latitude on different hemispheres, but the effect was different in different oceans, with a tendency for the effect to be greater in the Atlantic and Indian Oceans than the Pacific. So that's the overall summary of studies on the effect of latitude on marine biodiversity up until that time, 2004. It seems to be a regional effect that is not seen at local sites where local factors prevent it from operating. And what might those local factors be? Well, any internal dynamics related to competition and predation, or perhaps habitat, seem to be preventing a site from reaching the highest species richness that it is capable of experiencing based on the region it occupies and the total regional species pool. And of course, this is just the main effect of latitude on diversity. The study does not control for other effects that might be causing the diversity gradient that are just correlated with latitude. So as we saw in the coral reefs example, where the main cause of higher alpha richness was the amount of habitat area or shallow ground surrounding the sampling site, and this shallow ground is greater closer to the equator for all those equatorial oceanic islands. So given there's a large and significant effect of latitude on marine biodiversity with higher diversity near the equator, what might be the cause? Well, there are several possible explanations, and some or all might be operating, and there might be different causes for different habitats and species groups, and this is an active area of research. So here's a list of possible causes, just sort of, um, in theory, that could be operating. Let's talk about those, and then we'll look at a more recent study that summed up several of these possibilities. Okay, first, productivity. Higher or more constant and stable productivity, NPP, talking about primary productivity, might contribute to a higher diversity. So if total productivity is higher, then that productivity can be broken down into smaller chunks. Each of which can support a species separately. So if productivity is constant and stable, then environmental stochasticity might be less pronounced and populations are less likely to go extinct locally. All right, so high productivity is one way that you could possibly support a larger number of species. So you can break down each of these chunks of productivity and still be large enough to support a separate species and stable enough to prevent extinction. Net primary productivity, however, is not higher in the marine tropics. It's rather higher in any areas of coastal upwelling. So this would predict higher biodiversity in coastal upwelling areas, really, not necessarily in the tropics. Tropics does have most stable year-round productivity with less seasonal variability than the temperate or polar regions, as we've seen. So this certainly could potentially explain higher biodiversity in the tropics. However, it would seem to promote a local effect on biodiversity, not a regional effect, as environmental stochasticity is not as much of a regional as a local effect. Local populations might go extinct, but only locally, not regionally, caused by environmental stochasticity. So this explanation seems questionable, at least in part, as a mechanism for the observed large regional effect, although it certainly could contribute to the smaller observed local effect. So high net productivity and year-round productivity could be contributing factors. And that's the niche partitioning hypothesis, tropics. Second, speciation rate. Studies have shown that in many terrestrial lineages, speciation rate is higher in the tropics than in the temperate regions. And this may be due to the fact that the tropics are a more stable environment, have a larger land area, and hence more habitats, or have higher mutation rates due to higher UV radiation and temperatures. Okay, so not clear the extent to which this might apply in the marine environment. But and also, even though speciation rates might be higher, still these species need to need to be able to persist locally or regionally in order to have an effect. All right, so they need to survive the various constraints and filters of habitats and niche and biogeography in order to persist and be counted. Number three, we have area. Land areas greater in the tropics and certainly oceanic islands and shallow regions suitable for coral reefs are more abundant in the tropics and do explain the greater coral reef diversity near the equator. But it's not clear how or what type of greater area in the tropics would be able to explain higher diversity in other types of pelagic or benthic habitats there besides coral reefs. So this coral reef pattern does not necessarily explain all of what we see in higher diversity near the tropics at lower latitudes. Fourth, we have evolutionary age. The tropics have existed continuously over the last, 500 million years since the snowball Earth, whereas colder regions have a more variable existence over the history of the planet. Because the tropics have had a more stable existence over evolutionary time, the stable, warm environment required by tropical species has been more reliable. And this might explain why the rate of extinction is on average lower for species in tropical lineages than in temperate areas. Terrestrial land branches of the evolutionary tree appear to be less likely to die in the tropics. Tropics might be the museum of biodiversity. Ecological interactions, number five. Some have proposed that predator-prey arms races during evolution or character displacement in competitive species are major interactions promoting speciation. So this was one of Darwin's ideas. And because there are more of these interactions in the tropics, the speciation rate might be higher in the tropics. So this is just a version of the above hypothesis on speciation rate, really, and is somewhat circular, as you need more species to begin with to have more ecological interactions. But it's definitely true that ecological interactions, competitive and predatory interactions and arms races do contribute to and promote a high speciation rate. Then we have the mid-domain model. This is the idea that a species' ranges are chosen randomly within a larger bounded domain. Just choose a range randomly. On average, more species are going to be overlapping in their ranges near the center of the domain than near the edges of the domain. So the regional diversity or the gamma richness would be higher near the center of the domain purely by chance than near the edges. So if equatorial regions tend to be near the center of a domain of inhabitable habitat on Earth, then we'd expect more species near the equator purely by chance. So no explanation at all is even needed for the observation of higher regional tropical diversity if that's a mid-domain. The equator is in the middle of the domain of inhabitable space on Earth, so naturally we might predict more species there. So those are a sampling of some of the ideas that have been proposed to explain the higher diversity in the tropics, mainly terrestrial, but also they could apply to the marine in some cases, the marine environment. All right, so recently there was this big paper that came out that looked at some of these in a systematic way and tried to explain them, basically a rehashing of the data set, looking at what's happened since the Hillebrand paper came out. All right, so this was in 2017. So what do we have in this most recent study telling us the causes of this gradient in species richness with latitude? Okay, here we go. This is what these authors found. We've got site diversity, alpha, local diversity, also alpha, and ecoregion diversity, a gamma diversity. So the site diversity is just mean species richness per transect. The local diversity, or local richness, basically, See down here in the caption, they're calling these richness, but in the figure, they're calling them diversity, okay? So local richness are estimates of total richness for sites, you know, within a scale of around 12 kilometers. And then the geographical ecoregion richness is basically the gamma richness. And we're looking at all taxa and then separating out vertebrates and invertebrates. So what we see is the hotspot of diversity in vertebrates and to also a large extent in invertebrates is this Indo-Australian archipelago right here in the South Pacific, the Southwest Pacific. So this is a hotspot of diversity across all groups. And it's in the ecoregion, in the local diversity, and also in the site diversity, except for invertebrates. In sites in invertebrates, we have hotspots at the higher northern latitudes in the Pacific. So this is a Pacific hotspot for large mobile invertebrates diversity. And here's another picture of same diagram except broken down more specifically into different animal groups. And we have the same hotspot here, the Indo-Australian hotspot for the finfish, sharks, skates, and rays, and the marine reptiles, so the vertebrates, the marine vertebrates. Then if we look at the rest of the species, all invertebrates, it's quite a different story. Similar hotspot for the crinoideas, but for the crustaceans, it's the northern Pacific and Atlantic, and especially the North Sea, and even, look, the Adriatic Sea is a hotspot for crustaceans. Asteroidea, we have again the far north, especially the Pacific, Eastern Pacific, and South America. Echinoidea, again, the northern and eastern Pacific, especially South America. Got a little bit of the southern Pacific here, where most of the species are. Holothuroidea, mainly the eastern Pacific, wide range of latitudes and high latitudes up in the temperate northwestern Pacific. Gastropods, far north, Atlantic and Pacific, and South America. Cephalopods, mainly the northern Pacific, and a bit here around the equator on the eastern side of the Pacific. So not a simple pattern of higher species richness in lower latitudes near the equator. It's a bit more complicated. 4 species per transect. Also, the North Sea in general. Oh, okay, so we do these correlations, or sorry, these regressions of site abundance, site richness, local richness, ecoregion richness of species with latitude, going from higher latitudes down to equatorial regions. We find these interesting patterns. Generally a negative slope, sometimes peaking not at the equator, but somewhere near the equator, around 10 degrees latitude. For vertebrates and for all taxa combined, but for invertebrates, we get a different complicated pattern here. We've got negative slope close to the equator, but at the site level, again, we've got higher species richness in temperate and higher latitudes. And these represents large mobile species. Um, Generally higher numbers of those species within a site at higher latitudes than close to the equator. All right, how do we explain these patterns? Well, there's 1, 2, 3, 4, 5, 6, 7 hypotheses these authors tried to address with these data. And they tested these hypotheses using a regression model involving these predictor variables that are related to each hypothesis. So the temperature hypothesis was addressed with by regressing species richness with the sea surface temperature. The primary productivity hypothesis with chlorophyll, nitrate, photosynthetically active radiation with species richness at sites. The species area, or the habitat area hypothesis with the total continental shelf area near the site of sampling, and also the total area of coral reef within the ecoregion where the sampling site was. The regression of natural disturbance with species richness was done using the cyclone data set. The percentage of sites surveyed in an ecoregion with a cyclone, at least category 3, passing within 50 kilometers in the last 10 years. And they also looked at, tried to quantify disturbance with the range of sea surface temperature. Greater range meaning implying higher disturbance. So disturbance here basically being defined as density independent factors that can bring down populations. Climatic stability hypothesis was addressed with looking at the 1 over the standard deviation of mean water temperature since the last glacial maximum. Estimated for 3 time periods, 8, 21, and recently the time periods. Fragmentation and connectivity is a hypothesis explaining richness patterns. This was addressed with looking at the total length of the coastline, the log of that, the number of abutting coastal ecoregions, and the number of separated coastline features, basically the number of islands in the area. Then they also looked at human disturbance, so if that had an effect, and measured as the population, the human population size, density in that area sampled. All right, let's take a look at each of these and just think about them for a little bit, how they could be causing, how they could be related to richness, species richness. So how could temperature relate to species richness? Well, if you have high temperatures, then maybe you have increased metabolic rates, faster generation times, faster mutation rates, and faster speciation, higher speciation rates. So that's one mechanism whereby areas with higher temperature could have more species. Also, it might be that more body plans, more types of body plans are possible at high temperatures than at low temperatures. So high temperatures might be more forgiving to variation in the body plan. So they might allow more body plans or more species to be able to exist than in areas with low temperature. That might be one possible reason. Another possible reason is that temperature might be correlated with productivity. And so there could be a temperature effect through productivity. So if we've seen a higher productivity might lead to higher species richness just due to Resource partitioning. You can partition up more chunks of that resource if you've got more resource to begin with. And you partition up more of that resource, then more species can coexist on that partitioned resource. Remember, the competitive exclusion rule, the number of species coexisting at equilibrium has to be no greater than the number of resources that they are limiting resources that are within that community of competing species. So if you have a higher productivity, then competitors can divide up that productivity more, and so more competitors can coexist. All right, habitat area, independent of habitat diversity, increased area results in increased number of species at equilibrium. That's a basic result of island biogeography. All right, some mild disturbance might maintain species. Okay, so that would be similar to the keystone predator effect. So if you have some disturbance that is bringing down, so this is not a biological predator effect, but rather an abiotic effect. So you have cyclones or wide variability in sea surface temperature that might cause populations of dominant competitors to be reduced. So that might maintain competitors at low levels, preventing them from reaching their shutout levels and eliminating their competitors. So a disturbance might tend to reduce competition and maintain more species than areas that are not disturbed. Climatic stability, greater stability might result in lower extinction rates and therefore higher species richness. Fragmentation and connectivity. So the more fragmented areas are, the more likely that speciation can occur because we have lower rates of gene flow between areas that experience different habitats and different selection regimes. So habitat is fragmented, then maybe more species can evolve and we have higher rates of speciation in these areas. That's another possibility. Then of course human disturbance can cause extinction and reduce the species richness. Okay then, what are their results? What determines marine species richness at the site, local, and ecoregion scale? And how is it related to latitude? Okay, so here's a table of the results. We've got all taxa and then the individual groups here. Mean abundance at a site, richness at a site, richness at the site corrected for mean abundance. Local and ecoregion richness. And the bold indicates a significant effect of this predictor variable on this response variable, explaining this percentage of the variation in this response variable. So for example, sea surface temperature is significantly correlated with site richness in vertebrate species and explains 85% of the variation in site richness among sites. Is also positively correlated with mean abundance and local richness of vertebrate species. In invertebrates, we have a different story. Abundance and site richness is correlated with chlorophyll, so productivity. And local richness is correlated with the amount of coastline and the area of continental shelf. In both vertebrates and invertebrates, ecoregion richness is correlated with nutrients and with island biogeographical factors like the amount of coral reef area and the amount of continental shelf area nearby. Here's a nice diagram that shows these relationships a little bit more clearly visually. So first, temperature and nutrients both influence site abundance. Okay, so the warmer the surface waters, the more abundant species are, and the more nutrients in the surface waters, the more abundant species are. And abundance influences site richness. So the more abundant species are, the more species are at the site, the higher the alpha richness. And the alpha richness at the site greatly influences the local richness also at the site. So these two different scales of alpha richness here. However, there's a negative correlation between vertebrate and invertebrate site richness. Sites that have a large richness of invertebrates have a low richness of fish, and vice versa. So the authors hypothesize that the reason for this negative correlation, remember, the vertebrate richness hotspot is in the southwest Pacific, while the invertebrate richness hotspots are elsewhere. The reason for this, according to the authors, is that the fishes, predatory fishes, control abundance of large mobile invertebrates through predation. So where there's a high richness of fishes, you have a low richness of invertebrates because of that. And in other words, at that Indo-Australian archipelago, that high richness of fish results in, through predation, in a low richness of large mobile vertebrates. All right, at the ecoregion scale, species richness is influenced by local richness, also by the coral reef area and the amount of continental shelf in both vertebrates and invertebrates. So these are island biogeography factors that come into play at the regional level. And the coral reef area has a much greater effect on the ecoregion richness, or the gamma richness of a region, than the local richness at that particular site, at any particular site in the region, for invertebrates. And the two have about the same effect for vertebrates. Right, and here's a nice diagram sort of visually summarizing all of this. So we've got abundance, site richness, local richness, and ecoregion richness. So we can see the relationship to latitude. And we see in terms of abundance, fishes have the high abundance near the equator, and vertebrates have the highest abundance near the poles. This is a negative relationship between fishes and invertebrates globally, caused probably by predation by fishes on those invertebrates. Site richness, we also have a negative correlation between the number of species of fish at a site and the number of species of invertebrates. So fish have the highest richness at a site. At the equator, lower latitudes, where it's the reverse for invertebrates. They have a higher mean site richness at higher latitudes, north and south. And again, the reason might be because of predation by fishes on invertebrates, not only driving the abundance down, but driving the number of species down in areas where the fishes are abundant and highly diverse, many species. And that occurs within individual sites. That negative relationship occurs within individual sites, but it does not occur locally at over a larger area. That's because exclusion, predatory exclusion, occurs at the small scale at an individual site, but it does not occur locally over several sites at the same time. So the local richness doesn't really change according to latitude for invertebrates, but it does for fishes, and it's influenced in part by site richness. Okay, the ecoregion richness is affected by island biogeography factors like the area of coral reefs and the area of continental shelf nearby. And at the ecoregional level, the gamma richness, the fishes have the highest richness near the equator, but not quite at the equator, more, north and south, 20 degrees latitude, north and south. And the invertebrates are similar, because both of these are influenced by those island biogeography factors relating to area of coral reef and area of continental shelf, which is highest near the equator. Not necessarily right at the equator, but a little bit removed from the equator. So can we now answer that fundamental question about life on Earth? Why is species richness in marine environments correlated with latitude, with higher richness near the equator? What's the answer? Well, the answer is it's complicated and depends on what scale you're measuring richness at. So if you're looking at a site, an individual transect, well, the main factor that determines richness is temperature, and the tropics are warmer than the polar regions. So in the case of fishes, the highest richness occurs near the equator in warmer waters. But those fishes prey on invertebrates, so invertebrates have somewhat lower richness at the equatorial waters. Negative relationship between the two within sites at an individual transect scale. And if you widen your area of spatial scope to the local area, then temperature also has an effect, but island biogeography also has some effects. So the amount of coral area and the amount of coastline also determines the species richness. And those factors tend to increase near the equator. But again, there's probably an effect of fish predation on invertebrates, preventing invertebrates at the local scale from also increasing in their richness due to those same factors. And at the ecoregion scale, the gamma richness scale, we have again island biogeography factors like coral reef area and shelf area determining richness, especially of fishes, and these are greatest near the equator. And somewhat lesser effect for invertebrates, possibly for the same reasons, but still an effect at the ecoregion scale for invertebrates closer to the equator, but not right at the equator for either fishes or invertebrates. So in this study, although we get a correlation with latitude, it's not clear that this correlation is independent from the other factors. So latitude might not have an effect by itself. Rather, these other factors are the main reasons, the main causal factors, and they just happen to be correlated with latitude. So the main factors apparently causing higher richness near the equator are simply the fact that temperature is higher near the equator, and also the amount of coral reef habitat, and the amount of coastline, and the amount of shelf area are greater near the equator because of all of these archipelago islands here in the western Pacific. Also, nitrates negatively affect richness at the local scale. This should actually be over here. And as we know, nutrients are greatest in the poles and temperate regions, so that might also contribute to the higher richness near the equator. All right, so there's the answer to this fundamental question in ecology, at least as of April 2021. I'm sure research is continuing. It's a very active area on this, and we'll, I'm sure, see a lot of interesting studies coming up in the future. Still, we don't know what the precise mechanism is, how temperature acts to influence richness at any scale or abundance, and the role of nitrates either. But the effective coral reef area, coastline, shelf area, I think that's fairly well understood. Nevertheless, still a lot of questions left to be answered. Still a ripe and wide open area for very interesting research. All right, so this has been just a brief synopsis of the most important and active areas of research currently in the subfield of community ecology. And of course, there are many additional areas of research that we haven't been able to touch on really, including, for example, the relationship between community stability and diversity and the idea of ecosystem services, which you can find discussed in the papers and some of the textbooks linked on the Course Merlin page. And again, community ecology sits mainly here at the outcome, the bottom, at the outcome of the struggle for existence, the outcome and results in terms of the kinds of species, their functions, roles, interactions, their abundance, and the total number of species that we see within any local community as we're out on the beach snorkeling around and observing. All right, this has been the subject of ecology, marine ecology. It's a huge subject, and we've barely scratched the surface of the most important and actively researched areas at the current time. And as you all know and are experts about now, the field of ecology started with Darwin's book, Origin of Species, and specifically as chapter 3 on the struggle for existence here. And we've looked at this struggle now from every perspective and looked at the results from basic research, whose goal has been to shine a light on various aspects, all possible aspects of this struggle for existence. And the central insight of Darwin's is natural selection and fitness, a trait of an organism. This is the central concept and property that influences behavior and emergent properties of all biological systems at all levels of organization, organisms, populations, and fitness affects all these properties, often in predictable ways, sometimes surprising ways. And all these properties constrain organisms in all possible ways, influencing how they're capable of carrying out their life functions, realizing their fitness, whatever it may be. So we have this endless cycling back and forth between fitness and all other properties of biological systems as they constantly change over time as a result of evolution by natural selection on this watery planet that by some happy miracle has life on it. And I'll conclude these lectures by quoting the last paragraph, poetic paragraph of The Origin of Species. Okay, so Charles Darwin concludes, it is interesting to contemplate an entangled bank clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the damp earth. And to reflect that these elaborately constructed forms so different from each other and dependent on each other in so complex a manner, have all been produced by laws acting around us. These laws, taken in the largest sense, being growth with reproduction, inheritance, which is almost implied by reproduction, variability from the indirect and direct action of the external conditions of life, and from use and disuse, a ratio of increase so high as to lead to a struggle for life, and as a consequence, to natural selection, entailing divergence of character and the extinction of less improved forms. Thus, from the war of nature, from famine and death, the most exalted object which we are capable of conceiving, namely the production of the higher animals, directly follows. There's grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Thank you, Mr. Darwin.